Hello everyone. I hope you all can hear me out and see the presentation. Just confirm me if you can hear me out and see the presentation, guys. Just confirm me once. Anybody?
Yes, sir.
Okay,
It's
okay.
visible and audible.
Great, great, great. So this is the enrichment session 2, paper number 2, and we are going to see the revision and some enrichment of the most important unit of paper number 2. That is a plant breeding. Okay. I have told you that there are in paper number 2, there are total 7 units. And the most practical and rewarding strategy will be to have the hold over 5 units, complete hold over 5 units. The strategy should be this practical approach I'm telling you. And rest 2 units, which I'm right now telling you, you can just go through the syllabus headings also. Because always in paper number 2, you will be getting the choices. I hope you must be understanding this. So, in order to utilize the choices which are given, you have to have short done with these 5 units. The one remaining unit which is problematic in paper 2, which you also must be fearing, will be about pest and the diseases and the horticulture crops. That can be left also. Left means only few important pests and few important horticulture crops you will be doing and you're good to go. Don't worry about these components. But some components over here, some units over here should not be left by you. Unit 1 is already done and dusted by us. That is cell biology and genetics. Very important. Every year you will be finding minimum 2 questions from here. Maximum 3 questions from here. Now the next unit today's which we are going to revise is unit number 2, that is a plant breeding. Again, minimum 2 question, maximum 3. As per the exam nature, it will be coming. Minimum 2 question you will be spotting over here. In 2025 also, you will be finding 2 questions from plant breeding. Broad topics. Then unit 4 over here. And unit 3 and unit 6 will be coming over here inside your 5 units. 1, 2, 3, 4, 5. 5 units. I have already told you. Again, I am telling you this. Okay. So unit number 2, we are going ahead with plant breeding. Plant breeding over here, if we will see the syllabus, we have divided it in this form. Okay. But now, as this is a revision session, I'll not be focusing upon the less important part. That part will be doing in the end. I'll be focusing upon the more important part, means from where one question is fixed from for every year. Every year you are going to see one question fixed from here. Though the questions will be coming around 2 to 3, but one question will be coming from that subunit only, that subpart only. Can you guess which is that particular part from where question is coming every year and it will come in future also every year? What is that part? What is the line, that syllabus line?
Selection type.
Selection type, right, Satakshi? Okay. Over here, just wait. Yeah. So this over here, you will be finding pure line selection, pedigree, mass, recurrent. This is the line which you are seeing. Every year, you are going to find one question from here, guys. ठीक है, either in your state PCS or any examination you are giving with respect to agriculture, this is the most important part. So we'll be more spending in this particular time, like this particular enrichment session, revision session, will be more spending time for starting from here, and then we'll be covering the less important topic like that. And probably we'll be dividing this plant breeding into 2 parts, so there is no rush for us to complete it in one go, because we need to have the productivity also with respect to the topics. Okay. So we'll try to understand this pure line selection, pedigree, mass, recurrent selection, because you will be finding this is not a one pager information with this particular subtopic. Some topics like molecular markers application, you will be understanding in the end, and they are one pager only. Over here, you don't need much information, but this over here is a multiple pager information which you have to have. ठीक है, rest are you will be finding are interconnected, like breeding for disease and pest resistance is connected with unit number 6. ठीक है, and then role means application part is over here, interspecific, intergeneric hybridization. This we will be seeing in the end, right? So first, let us start with this pure line selection, because a lot of student must be facing problem with respect to this particular topic too. ठीक है. In order to understand this, let us first understand about the flower, right? And one topic which I have left was the heritability in unit number 1. So that also we are going to understand, guys. So before moving ahead, let us anatomically, let us draw the flower, because flower understanding should be there. Flower morphology you have to understand. It is very important. The sporogenesis and gametogenesis you have to understand. So with that, we will be starting and then we will be moving forward. There are 4 whorls in the flower. What are the 4 whorls in the flower, guys? Can you tell me the 4 whorls that are in the flowers?
The
Calyx, corolla,
corolla
sepals, petal,
Right. Over here, calyx, corolla,
and
androecium, gynoecium.
gynoecium.
androecium, and gynoecium. ठीक है, calyx, corolla, androecium, gynoecium. This you have to draw it in the examination also. Calyx is related to
Sepals.
Sepals,
corolla is related to
Petals.
androecium is related to
Stamen bud.
stamen, and stamen is containing your anther and filament.
Filaments.
Gynoecium, this is the male part, right? All
Yes.
this you have to write in the examination only. Gynoecium,
Female part.
female part. It is stigma, style,
Oil.
and ovary. Over here you will be finding. Okay. So this you have to draw it in the examination also, and I have told you you will be drawing it like in the examination too. That in the lecture I have told you to draw also. ठीक है, ये बनाना भी पड़ेगा आपको. Like this you can draw it in the examination also. यहाँ पर आपके कुछ petals you will be showing like this. Over here like this, some sepals you will be showing. This is the androecium, right? And over here this will be your ovary part, and inside that ovule. This is the gynoecium, this is androecium, this is petals, this is calyx, and then their pedicle also. Just leave that part. It's not very important. Okay. So this is one thing. Now over here, I'm stressing upon the fact which you should remember is the gametogenesis or sporogenesis. One is a microsporogenesis. Another one is megasporogenesis. This is something typical over here which you have to understand also. What happens in microsporogenesis, guys? What you will be writing if they ask you about this particular part over here? Microsporogenesis. What happens under microsporogenesis? Tell me.
pollen formation.
Microsporogenesis is responsible for pollen formation. And pollen is your ultimate gamete over here, which you will be getting. So how this particular microsporogenesis happens? In the lecture also I have told you. And this might be asked too, because it is given in detail in the books also. But a rough understanding should be there. So over here, you will be finding the stamen part, right? This particular anther you will be finding. Anther is bilobed or trilobed? Anther you will be finding bilobed. Okay, and when you are cutting this particular cross section over here, and when you will be finding the top portion, you will be seeing the sporangiate like this. From what pollen is getting formed? 4 pollen you will be getting over here. What is this called? Inside this we will be having. What is this called, guys? Pollen mother cell you will be finding over here, or microspore, like you will be finding mother cell over here. And from here, what is the ploidy of this particular PMC? Ploidy, pollen mother cell. And what is the ploidy of 4 pollen, which you will be getting over?
Haploids.
Haploid. So this is the diploid over here, 2n, And what you will be getting over here, the 4 pollens will be haploids. Okay, this you have to write also in the examination. Let me show you how you can write it over here. Okay, one thing is this over here, and megasporogenesis, what you will be finding? Megasporogenesis. Again, the megaspore mother cell. But where you will be finding that megaspore mother cell? Inside the ovary. Suppose this is the ovary. And ovary is connected over here. Ovules are connected with the placenta like this, you will be finding. Okay, there will be ovules. These are ovules over here. And when you will be finding these ovules are connected over here like this, you will be finding a ovule. And at one end, you will be finding this megaspore mother cell. Okay, which will be happening again 2N. Now this megaspore mother cell will be going into the formation of this meiosis process. And then I have told you that it will be going into the thrice mitosis will be happening. That's why you will be getting 8 nuclei. Okay, a rough understanding you have to have over here. Now let me show you actual what you will be writing it. Just wait. So this over here, see this guys. Okay. Like this, you will be finding the above interface of the anther. ये पूरा anther bilobe आपको देखने को मिल रहा है. Over here, you are finding epidermis, endothecium. If you want to remember it, you can remember, but otherwise, epidermis is the outer layer, right? Endothecium is like this inner layer you will be finding. Then over here, middle part is filled up with this middle layer. And over here, the most important part which you have to highlight also, this is sporangenous tissue. And a sporangenous tissue is containing your polar mother cell. Okay, so यहाँ पर over here you will be finding some information given about them. Again, in detail if you want to go. Otherwise, there is no need to. Because direct question till now has not come on this particular part only. They will be giving you a 2-part question again, right? So you will not be getting so much in-depth question that you just have to explain microsporogenesis, right? Then diploid nucleus you are finding inside that particular sporangenous tissue. Meiosis will be happening, converting into 2. Then haploid nuclear N will be formed. This is 2N over here. And tetrad will be formed. 4 microspores will be formed over here, which will be ultimately giving the pollens over here, right? So this will be the formation of pollen. Now pollen, this is the outer layer of pollen you will be finding called as exine. This you have to remember, guys. Exine. Okay. And the inner layer you will be finding as intine. Inside this cytoplasm. Okay. Over here you will be finding. But the most important part is I have told you first initially there will be one vegetative part and one generative part. Right. Generative nucleus will be there and one vegetative nucleus will be there. Then this generative nucleus will be converting into 2 particular gametes. And this vegetative part will be responsible for the formation of pollen tube over here. Or it will be guiding the path. So like this, you will be finding the vegetative cell part. Like this, you will be finding. And the generative cell part. These are NCERT images of 12th class NCERT. If you want to go through that, you can go. But a lot of information is written over there. So selectively understand it. Okay, because the most important part is not this from this particular unit. Okay. So over here you will be finding, okay, this you have understood that this is the generative cell, this is the vegetative cell. Generative cell will be dividing into 2, and then you will be finding 2 nuclei over here, and these are nothing but your gametes which are ready to go into the over here stigma part. Okay. Now this I have told you 4 tetrad. This is a generative cell, this is pollen wall, and then sperm cells will be generated from the generative cell. So mitosis of generative cell is happening over here. This is N, so 2N will be converted. Okay, so this is going to happen over here, guys. Now this is the part of your female reproductive part, stigma. and pollen will be landing over here. Okay, and from here that this vegetative part will be going like in the lecture I have told you already, and that thing will be happening. But separately, if they will be asking you about the microsporogenesis or megasporogenesis, then you have to draw a bit. In one page, you have to complete this, right? So these are the stigma style. I have told you that this is the placenta and ovule is connected like this, like this. Okay, right now, because in the lecture I have told you directly that this type of formation I have told you directly, right? But this type of formation inside the ovule happens over here. Suppose this is one ovule I have taken. So over here it will be happening. Okay, so this, if I am opening it up, this is the outermost chalazal. This is nucellus, outer integument, inner integument. This is part is inner integument, and this is the micropyle end. Okay, this is funicle and this is connected with placenta. Now at the micropyle end, what you are finding over here, this nucellus is going to form a megaspore mother cell, MMC. And from this megaspore mother cell, you will be finding that meiosis is going to happen. Again, because of meiosis, there will be 4 daughter nuclei which are going to form. See over here, if they have showed you, right? Megaspore mother cell, this is nucellus. From here, you will be finding that there is a megaspore tetrad which will be formed. These are the 4 parts. Do all these 4 participate in the formation of an ovule or in the formation of the egg cell? Do all these particular participate, guys? Can you answer?
No, sir.
Not all participate. So what happens to the rest of the 3?
It will degenerate.
It will degenerate, right? So this will degenerate over here. These parts will be degenerating over here, and one will become functional megaspore over here. And after this, you will be finding there will be mitosis, which is going to happen. One mitosis, second mitosis, and third mitosis. In the third mitosis, you are going to find the 8 nuclei, you can say. In the NCRTs, they have shown you this particular cells, that there will be 7 cells which will be created. 1 cell, 2 cell, 3 cell, 4 cell, 5, 6, 7 cell. So inside the middle one, you will be having 2 nuclei. Cell or nuclei mein difference hai. There is a difference between cell and nuclei, right? And over here, you will be finding this type of arrangement, which I have directly shown in the examination or in your lecture also, and you will be directly showing it too. Because I don't think so, again, that they will be giving you a full-fledged question on megasporogenesis. Till now, they have given. Till now, they don't have given it. Okay? So like this, the formation you will be finding, which I have shown you. This type of formation, which I have shown you. Exactly, you can remember. The middle one is Excel. This is your synergies. These are antipodal. These are 2 polar nuclei which are going to find. This ultimately you have to remember too. Right, chalazal and micropylar end. So ultimately, I wanted to a bit explain you this, that how this things is happening over here, and then you will be finding the pollination part will become and fertilization will be happening over here. Okay, so one thing you will be understanding with this particular part. The most important part which we are going to see now will be your selection. But before going into the selection part also, tell me about the heritability concept. Heritability. Just wait a minute. Yes, guys, heritability. In this particular part, I might not have taught you in unit number 1. Heritability. What is this heritability, guys? What do you understand by heritability? And direct, like rarely they have given question on heritability, but one time they have mentioned it. So what do you understand by heritability? Tell me, anyone. बताओ भाई, what definition you would want to give, or what understanding you have with this respect to heritability. And after that, the question will be, what do you understand by heritability, and what is the difference between the broad sense heritability and narrow sense heritability. This is how this particular question is going to come, and it has come also. Where it is written that heritability, where this particular part is written in the syllabus, can you tell me that first of all?
Variation of traits.
Okay, definition you are saying are variation in traits, heritability. देखो, don't go by the like how variation of traits is happening. Variation of traits. How this variation is happening, but heritability, you don't go by the exact meaning of this word. If you will go by that, then you might be getting confused. Over here, certain words should be included. Can anybody tell me if you want to try? It is a way heritability denotes, or it is basically, heritability is the proportion of the total proportion of the total phenotypic variation in a population that is due to what? Genetic factors. Understanding, this is the exact definition which you will be understanding, which I'll make you understand this too, because this is by concept, it is not complicated, but by this particular the words which are written over here, it becomes a bit complicated topic to understand. Basically, what they want to understand is that there is variation happens in any organism you will be finding. Okay, so that if I donate, if I write it down like VP, phenotypic variation, that is visible. Now this phenotypic variation can happen by which things? What do you think? This phenotypic variation can happen by what all factors? Tell me that. बताओ भाई, किन किन चीजों से आपको देखने को मिलेगा ये phenotypic factors?
Genetic environment.
Right. First thing, you can get it by genetic. So I'll be writing the variation by genetic can cause phenotypic variation. That can happen. Just wait, na? Just wait, guys, na? Just wait. Just give me 5 minutes. There is urgent work. Just wait for 5 minutes and I'll be coming again. Just wait. Really sorry, guys. So where were we? I was telling you about the phenotypic variation. So this phenotypic variation might be happening because of the genotype. What else do you think?
Environment.
One thing, it might be happening from the environment. This is called environmental variation, right? So this you might find over here too. One more thing, because of one more thing, it might happen. And that is बताओ. This is phenotypic variation. Genetic. Environmental. As it is, you have to write in the examination also. VGE. That is genetic plus environmental condition. Right? And what do you like? How can you say this? Can you give any example? Like in good environment, gene have the best. You will be finding that they will be expressing itself the best. So environment also matters a lot. So there might be a combination of environment and genetics, and it is giving it is giving its effect in the progeny. It might be happening that it is not, the variation is not due to the genetics, but due to the environment. The environment which we are living, because of that certain variation can come, environmental variation. And then there is genetic variation. That is primarily because of genetics only. So what you are understanding, this is the phenotypic variation. Now you read the heritability definition again. Heritability is the proportion of total phenotypic variation that is due to genetic factors. यहाँ पर जो आपको देखने को मिलता है. But now you understand that over here there is some formula written for this heritability. And what is that formula which is mentioned for heritability? What is that one formula which you will be finding? The formula which is being written is VG upon VP. This is a one formula you will be finding, guys. That means the phenotypic variation, because of this genetic part, this will be giving you the H part over here, and numerical they can also give, but that will be a very simple numerical with respect to this. How they will be giving you, I'll be discussing in short while. But you have understood this. VP is equal to VG plus VE plus VGE. Combination of both. Now this VG genetic factors will also be having certain differentiation, guys. This might be this genetic variation which is showing, it might be because of certain things. So what you will be finding over here, इसको कैसे divide करोगे? Gene effect के basis पे divide करो. How the gene is interacting and how that gene can interact. Can you give me an example over here, or you must be knowing about this? There will be the additive effect of that particular gene. So over here, VA, additive means addition of a particular gene over here. That is additive. That one gene and second gene, they will both, their effects will both be adding together and will be giving you a summative or sum of these two. This is additive. There is a dominant one over here. That the dominance, like capital T, small t, and capital T will appear. Dominant effect. So this is a VD. Under VG, I am telling you this, VD. And then there is one more, that is epistatic effect over here. It is nothing but what over here, it is about two genes. Suppose this is A gene, suppose this is B gene. In epistasis, you will be finding that one gene will be masking the effect of the second gene. So when this is there, it will mask the effect of this. So this you can find over here, epistasis. Okay, VEP, you can add it like this. So VG is further divided into VA VD and epistasis. Understanding? So this you can write it in form of VG also. So this becomes your one more equation. Again, I'm telling you one question. If you will be seeing in the examination, you should not be shocked. They can provide you numerical on this basis also, and numerical will be much simpler. If you only know this and this equation, any type of simple numerical they will be giving you and they will be asking you to solve. ठीक है, ये देंगे कैसे वो मैं बताऊंगा. I'll be telling you that. So these two things I have told you, that VP and VG. Now you try to understand over here that if I'm talking about this formula, H square is equal to VG upon V. That means what you are understanding over here, that the total amount of phenotypic variance which you are seeing, how much is given by VG, that you are going to find. If I write VE by VP, what is this showing? That the amount, the contribution of environmental factor in VP. VGE upon VP, what it will be showing? The amount contributed by VGE in the phenotypic variation. तो इनका अलग अलग भी निकाला जा सकता है. In a broad sense, heritability, what you will be finding. Now there are two types of heritability. One is a broad sense heritability and one is a narrow sense heritability. In a broad sense heritability means that I want to take all the combination, all the types of genetic variation, all the effects of genetic variation with respect to the phenotypic variation which I'm being shown, like over here, कि एक particular species में what type of phenotypic variation is there because of genetic variation. Now I've told you that genetic is further divided between three things, three effects. So I'm taking all those effects into picture. So what my equation will be converting into, if I open up VG, how my equation will look like. So if I want to open the VG up, how my VG will look like over here? What should I write over here? Tell me, guys.
Heretic plus dominant plus epistasis variation.
VA,
VD,
VD,
VEP.
VI और EP आप लगा सकते हो यहाँ पर. VI, EP, जो भी आप लगाओ, whatever you will be writing over here, just give their index also, that this means this and this means this. ठीक है? VI or VEP you add over here. That is your broad sense heritability. That means broad means it might be of additive, it might be of dominant, and it might be of epistasis too. Overall genetic contribution to phenotypic variation. ठीक है? So over here you will be finding this broad sense. And if I write you the narrow sense, this is broad sense, guys. Okay, broad sense. Now if I say that my value of H is coming 0, what do you understand by this? If my value is coming near about to 0, and in the examination if they will be forming one numerical, and as per that numerical your value will be coming near to 0, what do you understand? What interpretation they will be asking to you? Or what interpretation you will be making if you will get 0 as your heritability?
Genetic variation is very less,
Yes,
close to
that's
0.
right. Very nice. Because 0, when this 0 can come, this H square, when it can come 0. Only it can come 0 when the numerator is becoming 0, because denominator cannot be 0. It will become infinity, right? So when it is coming near to 0, then only this particular heritability will be coming 0. That means is the numerator is coming 0. That means it is near about negligible. And if it is negligible, then it is showing me that the phenotypic variation which is coming is not because of genetic variation or genetic effect. It might be of environmental. This is giving you one interpretation. If my H is coming near to 1, what interpretation you will be having, guys? अब अगर मेरा numerator or this particular value is coming near to 1, what interpretation you will be having? Tell me.
All of phenotypic variation is due to genetics.
Right. That means all of phenotypic variation is because of genetics. How you have come to this conclusion? Explain.
VG equals to VP.
Vg will be becoming to Vp, right? Because then only it will come equal to 1. Or you can explain it like this also, that Vp is equal to Vg plus Ve plus Vge. So in order to make this equation equal to 1, what you have to do? In दोनों को, this part should become 0. Right? When this will become 0, automatically Vg by Vg will be happening over here. That is why Vp is coming to Vg. Because there is no effect of environment then. All the variance which you are seeing is because of genetics. Gene की वजह से you will be finding. That's why it is coming 1. And if it will be coming between 0 to 0 to 1, suppose 0.5 it has come. What inference you will draw from here? Tell me, if it is coming any value in between this 0 to 1, what inference you will be drawing over here? अब तो समझ में आ गया होगा ये.
Variations due to both genetics and environment.
That means there is effect of both genetics and the environment. So the game is all about over here with respect to the variation in a phenotypic with respect to genetic. That's why it is heritability, and this formula is over here. And with this formula, they will definitely be framing the numerical if they would want to. And every year now you are finding that one numerical is coming out of anywhere. A very simple numerical if you know the basic formulas. I have also already told you to. So this is broad sense heritability. Now, if I ask you, just wait a minute. If I ask you about this second narrow sense heritability, what do you understand by narrow sense heritability? What should come into the formula now? Narrow sense. Tell me, guys. Narrow sense heritability, by the word itself, you are saying that now I want to check narrowly. I want to check only one part over here. That means now Vg will not be there. Phenotypic variation will always remain because you want to check that how much variation of a particular thing in the phenotype you are seeing. What contribution you are finding over here. So now over here you will be writing, just wait. You will be over here mentioning about the A part. And what is this VA? That I want to know what is VA.
Additive.
Additives.
Right, additive variance. That means in a VG also, I am not concerned about dominance. I am not concerned about epistasis. I am concerned about the additive factor. And when I am taking this additive effect, that how much phenotypic variance has come because of this additive part, then it is becoming your narrow sense heritability. This is coming as a narrow sense heritability. Understanding? Because I am considering only additive genetic variation, which is directly transmitted from parents to offspring. Simple, you will be understanding this, guys. Okay. Now let me show you the content which you will be getting over here in the PPT. Just read that out too. Just wait. Okay, this heritability you will be getting over here h square Vg. But remember the way I have told you, that will be really helpful for you to understand it. First of all, Vp is equal to, draw this equation, Vg Ve plus Vge. Then you try to open this up also, Vg is equal to Va, Vd and Vi. And then you come ahead with this particular broad sense heritability and this broad sense heritability and this narrow sense heritability. And the interpretation, likewise, I have told you, they can mention you as per the percentage also, 0%, 100%, 50%. In percentage form also it can come. But generally will mean 0 to 1 and 0.5 in between these. The interpretation will be different over here. I hope this you have understood over here, guys. Okay. So this is one thing which I wanted to add up in your knowledge. Now we will be going to the important part over here, and that is with respect to your selection part. पहले वो complete करेंगे energy के साथ. Then we will be moving ahead. Just wait. ठीक है guys. So there are various methods of our over here the plant breeding. Now we are understanding the breeding methods over here. Okay, and in breeding methods you will be getting a lot of methods over here. First of all, like in pure line selection, what is written in the syllabus line will going by that point only. Pure line selection, pedigree, mass and recurrent selections, combining ability. In this combining ability, you will be understanding about GCA and SCA. And there will be backcross. Backcross is not written, but it has come in the examination. So backcross method also will be understanding. Okay. These all things will be understanding over here with respect to our objective. Our selection will be changing. Our objective or hybridization will also be changing over here. So first I am going to start with the recurrent because this is one of the difficult one, and we in the basic classes we have picked it up in the end. So there might be a rush for this particular topic too. Recurrent selection. Okay, pure line and everything you must be reading it again and again. I want to know about the recurrent selection right now. First of all, the objective of a recurrent selection over here. What is the objective of a recurrent selection, guys? Tell me. Why do you do this recurrent selection? क्या ऑब्जेक्टिव है रिकरेंट सिलेक्शन? बताओ भाई.
कोई भी अगर डिजायरेबल एलील है
राइट.
उसकी फ्रीक्वेंसी बढ़ाने के लिए.
Very well said. One particular part you should remember that recurrent selection is being done to increase the frequency of desirable alleles. This is the word which is written in your BD Singh as it is. 3 lines are written in BD Singh. BD Singh is one good book for this plant breeding, but again, read it selectively, or if you are following the lectures, you don't have to follow other thing. Try to focus more on rectification of the same topics. A recurrent selection means something we are selecting again and again, and we are selecting our variety again and again. So that means if we are selecting our variety again and again, there must be some purpose with that. That means this particular variety is having certain important traits which we want. But now we want to increase the frequency of that desirable alleles. That's why we are going up with this recurrent selection method in plant breeding. It is a cyclic breeding method. Also, you will be finding that we will be doing certain genetic variability also will be over here. Genetic variability will also be involved. Unlike the progeny selection you will be finding, or pure line selection you will be finding over here. Certain genetic variability will also be there. I'll be telling you. And we will not be focusing more on the inbreeding. So we'll ensure that minimum inbreeding should happen over here. So these things you will be understanding in a recurrent selection. Recurrent selection happens in majorly in self-pollinated crop or cross-pollinated crops. Tell me, guys. A recurrent selection happens in cross-pollinated crops or self-pollinated crops. It happens primarily in the cross-pollinated varieties over here. Cross-pollinated crops, you will be finding this. Okay, recurrent selection over here. Now, how recurrent selection happens? First of all, recurrent selection is of different types, which is very important. And it will be mixing your combining ability over here also. Recurrent selection. So recurrent selection happens. One simple recurrent selection might be there. This is simple recurrent selection. There might be a recurrent selection with general combining ability. So over here, recurrent selection with general combining ability.
Specific combining ability.
Great. SCA, specific combining ability. And one more is there. That is reciprocal recurrent selection. And this is recurrent selection with specific combining ability. SCA is specific combining ability. Okay, so these are the variants of this recurrent selection which you have to understand over here. Okay, this one thing you should know. Second thing, one by one we will be seeing it, but when this combining ability concept is coming over here, so first we will understand about this combining ability. What do you understand by this combining ability, guys? So what do you understand by this combining ability? If I ask you, what is the understanding of this combining ability? What do you understand by it? If in the examination it will be asked, how you will be writing this? Combining ability. And directly, this is one of the syllabus topic you are going to find. Yeah, they go combining ability hai. And they have asked twice from here. Difference between GCA and SCA. Directly it will be coming. Tell me. So combining ability, something is getting combined. What genes are getting combined only? So combining ability is the ability of a parent. Ability of a parent to pass on desirable traits. Ability of this parent to pass on desirable traits to its offspring. What we are checking over here, the ability of a parent to pass its desirable traits, how well it can pass its desirable traits to its offspring, when crossed with other genotypes, it is known as a combining ability. So if we know a particular crop that the combining ability of this crop is poor, so why we will take it in the breeding process? We have to take a particular crop which should be having good combining ability. Now further we will divide it with two things. Combining ability will be dividing over here. Simple definition you have understood. Ability means the ability of whose parent to transfer what desirable traits to its offspring when crossed with other genotypes or other parents. Over here there are two types, that is general combining ability and specific combining ability. In a GCA, what do you understand by the GCA, guys? General combining ability. What do you understand by this? बताओ GCA क्या चीज है? What is GCA? Tell me, guys. देखो, GCA is general combining ability and specific. General means it must be about general thing, right? General over here. That means it is the average performance of a parent in a hybrid combination. For example, there is one plant I am having. I want to know its combining ability. I am saying right now combining ability. Combining ability, if I want to know, that means I want to understand that how much genes, how much desirable traits it can transfer to its progeny when combined with different genotypes. So suppose I am taking one A genotype, B genotype, C genotype, D genotype over here. Now I am crossing it with this particular X with A and I am getting a particular variety. And suppose I wanted over here, you can say that I wanted the yield. I want to check the yield. So over here, I am getting 10 gram yield. Example, with respect to A. I am getting over here with respect to B. Suppose this is giving me 8 gram yield. This is giving me 6 gram yield. And ultimately, we have got 10 gram yield over here as in the progeny. With respect to B, I am getting 11 gram. With respect to C, 9 gram. With respect to D, 12 gram. So when it is giving average performance of this particular X with respect to these genotypes, different hybrid combination, then I can say that it is having a general combining ability, GCA. Because over here, we are primarily focused upon the additive effect of the gene. And we are seeing that, okay, it is giving me on an average performance with respect to this particular two parents, earlier two parents, X and A, X and B, X and C. So this is general, right? Whereas if I'm talking about specific, so in specific, what I'll be doing? In specific, I'll be focusing upon that this particular X is performing really good with what parent over here. It becomes specific over here now. That with 11 gram, I am giving outstanding performance. So it is the deviation in performance of a hybrid from what is expected based on the average. Average, suppose 10 gram is expected, or average 8 gram plus 6 gram, 14 gram, इसका average क्या होगा? 7 gram. Average should be 7 gram. In GCA, all of the parent, when it is combined with, it is giving above average performance. But when it combined with the B parent, it is giving me 11 gram. That means outstanding performance over here, above average performance over here with respect to the GCA. So that means somewhere over here, which gene interaction must be at work, the dominance one or the epistasis one. So this is your specific combining ability. Let me show you the content. Just understand this much for now, then you will be understanding the recurrent selection really well. Okay, let me go to this way. So combining ability over here refers to the ability of parent to pass on desirable traits to offspring. Two types, GCA, average performance of a parent in hybrid combination. What it will be measuring? Additive gene effects. Right. And then over here, if you will be finding SCA, it is a deviation in performance. What is hybrid from what is expected on an average over here? It measures the non-additive gene effects like epistasis and dominance. Okay, so a rice hybrid from parent A and B performs far better than expected based on their individual GCA. This is high SCA over here. So this you will be finding with respect to this. This is a specific combining ability and the general combining ability. Now I want to understand about this particular simple recurrent selection first of all. What I'll be doing in simple recurrent selection, guys? What will be happening in simple recurrent selection? Tell me. What I'll be doing over here? Simple recurrent selection. So I'll be taking a population over here like this. You will be showing in the examination too. Okay. This is the variety which I have taken over here. And I want to improve the traits of this variety. Okay. These are the desirable traits which I need in this particular variety over here. So what I'll be
Desirable
doing?
traits are selected and self-pollinated.
Right.
In simple.
Again, Arya.
So desirable plants are selected and self-pollinated in simple recurrent selection.
Right, over here. Okay. What you will be doing over here, what you have said it rightly, that you will be now specifying over here. What you will be doing with respect to, suppose this is the X variety and you want to improve the desirable traits of this X variety by this simple recurrent selection method. You have grown it on the field like this. On the test field, you will be growing it. When it will be grown, what you will be doing with this particular part, you will be basically selecting the superior plants over here. Superior plants will be selected. Based upon how they will be selected on phenotype, based upon the phenotype, right? Phenotypically, it will be selected by what you are seeing. you will be selecting that particular traits. Good height, good yield, good particular, you will be finding whatever, there will be a list of traits which you want in your plant that you will be finding over here. Then what will be done? Suppose you have selected 10 plants out of it. Then what you will be doing? अभी खत्म नहीं हुआ है recurrent वाला. What you will be doing then?
Sir, again selfing.
Right. In this process, these 10 plants, now what you will be going ahead with, you will be making self-pollinate the selected plants. These 10 plants will be going to the self-pollination. This is very important over here, guys. Self-pollination will be happening over here. Self-pollination of the selected plants. ये आपको लिखना भी है. Like in the examination, you have to write this too. Then what you will be doing over here? So suppose there are 10 plants over here, like this. From this plant, you will be getting now seeds. You will be collecting seeds. Suppose from each plant, you are getting 10 seeds. So you will be collecting all these seeds over here. Seeds will be collected over here. Harvest seeds over here. Okay. Third part will be that you will be harvesting the seeds over here separately. Separate should be there. Separately, seeds should be harvested. All right. And then seeds will be evaluated for superior, like superior seeds you will be having over here. That what is the superior seeds over here? Harvest seeds and grow progeny rows over here. Like how you will be doing, like in the next. So what will be transferred in the next phase, in the next year, what you will be doing with this particular? You will be growing these particular selected seeds over here, selected plant seeds over here. Okay, that will be done by you. Now you will be evaluating the progeny performance. This progenies which we have added over here, we will be evaluating it. What I have done, individual plant progeny also we will say, because this is one plant, this is second plant, this is third plant, this is fourth plant, fifth, like that. 10 plants I have selected, 10 plants I have planted over here like this. How? Because from each plant I am getting certain seeds. So suppose 10 seeds I am getting, to 10 plants will be coming. 10 plants will be coming. Over here 10 plants will be coming. Okay, for the non-agri students, I'm making you understand this. Like this, it will be shown to you. Now over here, intercrossing will be happening. Intercrossing of all possible crosses means now over here, selfing in the first stage, we were doing the selfing. In the second stage, there will be crossing can happen over here in the project, in this particular plant also, with respect to each other they can have, or with respect to other plant also they can have over here the crossing. Right, and this is maintained while doing this plant breeding, and you don't have to go into this particular thing that how we are doing this particular intercrosses of all possible crosses of these plants. Okay, this is the, you will be finding in plant breeding in this particular domain, they will be making certain conditions that every plant should be able to cross with this particular plant also and other plant also over here, the progenies of other plant also. So objective is what you will be writing over here, that that over here all possible intercross will be made over here. Okay, all possible intercrosses will be made. Okay. And then what you will be doing, you will be selecting over here, you will be taking the seeds over here from all these particular crosses which you have taken. Yeah, so seeds you will be taking from all the crosses you have made, and then you will be mixing them up again. Again, you will be doing the mixing of over here. So that means now you have the seeds of the superior quality of plants in the population which you are having originally. So this will become your first cycle that you have performed this particular part, right? So now you will be performing the same action. What you will be doing, again, you are having a mixture of this particular seeds. Again, you will be selecting the phenotypically superior plants. Again, you will be self-pollinating them. You will be separately sowing them. And then again, when you are making the intercrosses, you will be again mixing them. Again, recurrent selection will be happening. Second cycle, you will be going ahead. And like this, you will be going to further, like recurrent cycles will be happening, like first, second, third, this particular recurrent selection cycle will be happening. And ultimately, you will be getting the desired result. How that over here you are basically have maintained the heterozygosity also, and you have increased the frequency of favorable alleles too. Okay, so this is one part. Let me show you how you will be writing it in the examination. ये BD Singh के diagram है, you don't have to go to this. In recurrent selection, you will be finding over here a simple recurrent cycle AR1. Like this, you can show it also by the schematic diagram. Select several superior plants based on phenotype. Self-pollinate the selected plants. Harvest seeds and grow progeny rows. First recurrent cycle which is going on. Evaluate progeny performance. Select the best progenies. Intercross. All intercross you will be making. Mix equal seeds from all crosses to form a composite population. And then from year third, again, you will be doing the selection, selfing, evaluation, and intercrossing over here. Then again, third recurrent cycle selection you will be doing. Features is, again, you have to remember we have increased the frequency of favorable alleles. Also, over here, why I am saying that heterozygosity will be maintained, because over here we are going ahead with the making the crosses, intercrosses amongst the plants too. Okay, that is also happening over here. So gradual genetic improvement, minimum inbreeding, and preserve genetic variability, and whatever traits we wanted, it can increase the effect of that traits too. This is your recurrent selection. Then why we are doing this GCA? Recurrent selection for general combining ability. What we are doing in this particular part, guys? Recurrent selection for general combining ability. What is the purpose of this particular doing this particular part, recurrent selection for genetic combining ability? Over here, we will be able to find, for example, if I want this my species to have, if I am finding it for good general combining ability, I wanted to know that this particular species should be good for most of the crosses if I'll have it with. All right. So this will be the objective of it, that I want to improve my population. I want to improve the yield and agronomic traits over here with respect to the population. So that's why we are doing, I am doing GCA. But if I want to make this particular variety good for a specific variety, when it will be crossed with a particular variety, I'll be going for SCA, general, this specific combining ability. So in this particular GCA, what should be the schematic diagram? What changes will be brought up over here, guys? So these are again your original plant population. What you will be doing over here, guys? Tell me. What changes is going to happen over here? Or what you will be remembering? Batao bhai.
The test class will do in.
Yes, we will be including over here a tester. A tester will be included over here. Okay, so over here you will be writing one tester will be included. Importance of tester. Now, why tester is needed? Can you tell me? Why tester we are including? Tester is nothing but a plant, male plant you will be finding over here. Okay, like this. Of different varieties you will be finding over here. Why tester is being utilized over here? Tell me, guys, tester क्यों चाहिए? Why tester is needed? Why I need over here some plant open pollinated variety? देखो, tester is having various over here variety, various types. So either I can have this open pollinated variety or inbred lines, which should be favorable for me to take for this GCA. Either I should go for OPV or inbred. What should I go for? OPV means open pollinated variety. Open pollinated variety. What do you understand by open pollinated variety? For the non-agri, I am explaining this. Open pollinated variety means that this plant you have left in the field, now this cross can happen with any other plant. You are not controlling the particular plant pollination. You are not letting it happen, that particular thing, what do we say, selfing, or you are not controlling the cross with one particular plant. So that is open pollinated variety which you are having over here. That means you are having different genes over here with respect to a singular. If you will take this plant, different gene combination, this plant, different gene combination, because this is your open pollinated variety. But if I'm saying that this plant has been generated by inbreeding, what do you understand by this? Inbreeding means all these plants will be having the same genetic composition. So if I want to check the general combining ability, I should go for OPV or inbred. What do you think?
OPV to check the combining ability with different plants.
Right. General combining ability you want to check with respect to different parents, different genes. Different parents mean genes. So different genes you will be not be getting in inbred. You will be getting in OPV basically over here, open pollinated variety. So you will be taking tester as open pollinated variety. Now this is the first part which is going on first year. You will be showing it in a different field. Original population you are having like this. Now what will be happening, guys? What you will be doing in this particular part? Tell me. बताओ भाई, बताओ, try to understand how you will be doing this particular GCA recurrent selection. Tell me, guys. Similar in simple recurrent selection, what you will be doing, you have to do it over here too. Just wait. You have to do it over here too. And what you will be doing over here, that you will be selecting, again, selection will be there, selecting superior, phenotypically superior plants. Suppose I have selected 5 superior plants over here. 1, 2, 3, 4, 5. Okay, 5 superior plants I have selected. In simple recurrent selection, what I was doing, I was growing their individual plant progeny. I was growing their seeds, and individual plant progeny was there, and then mixing their seeds after having the intercross. But over here, I'll not be doing this. What I'll be doing instead, when I have selected these plants over here, now I want to check this plant, all these plants, 1, 2, 3, 4, 5. Its general combining ability also I want to check. So what I'll be doing, I'll be now crossing them with these test crosses over here, guys. So I'll be doing the cross of each selected plant with the broad based tester which I am having to generate test cross progeny. So that means over here, one field which will be created, that will be having my test cross progenies. Right. So one is being crossed with these testers, second is crossed with these testers, third, fourth, fifth, and they will be grown over here. Okay. So this you are finding that, okay, I have gone ahead with the test cross over here, and I will be growing this test cross in this particular field. And I have this selected superior plants also. Now what I'll be doing in this test cross progeny? क्या करने वाला हूं मैं test cross progeny में? Tell me, guys. Over here, automatically, obviously, I have to select the best performing progenies. Best performing progenies will be selected from here. Suppose 2 plant has given me good result, 4 plant has given me good result over second plant, and 4th plant has given me good result. So I'll be selecting that individual plants over here. So I'll be identifying the best performing test crosses corresponding to the original plants which are superior GCA parents, means which are giving me superior result than the original population, nothing else. ठीक है. Now suppose I am getting, देखो इस टेस्ट क्रॉस से देखो ये बहुत ही इंपॉर्टेंट है. Over here just try to understand. There might be some confusion which might happen. On the basis of this, I have found, on the basis of this test cross, I have found that parent 2 or this particular 2 or 4 is giving me the best progenies, is giving me the best outcome. So what I'll be doing, I now will be selecting this 2 and 4 plants and doing the selfing of these plants. So I am doing the selfing of the originally selected plants, not the test crosses over here. Test cross I have done to identify that which of the plant is having better combining ability with the tester. And because of this, I have got to know that 2 and 4 is giving me better results. So now I will be doing this particular part, that 2 and 4 progenies I will be growing, and then I'll be making the all possible crosses. I'll be taking the seeds or mixed seeds of these particular 2 and 4 and going for the other recurrent cycle. I hope this you have understood, but let me tell you also by the write-up also how you will be understanding this. ठीक है, समझना important है, otherwise it will be difficult for you to understand this. See over here. I have to select the superior plants from the base population. So you will be drawing these two things. Base population means original population, tester. Cross each selected plant. So over here, each selected, suppose 1 and 2 plant I have selected. I will be testing it with the tester. So second air, you will be just testing over here with the test cross progenies. Over here, progenies will be coming. I will be evaluating the test cross progenies for yield, whatever traits I want, desirable traits. And I have got to know that these are the plants which are basically best performing with the test crosses. So suppose both of them are performing good because I have taken only 2. If I'm taking 5, I can select 2. Identify best performing test crosses. Corresponding original plants are the superior GCA. Isko chhod do. What it means that out of these 5, suppose I have 5 and 2 are performing really well. So what I'll do now, I'll be doing the selfing of these 2 only. And if I ask you what is the difference between this simple recurrent selection and recurrent selection with general combining ability in this step, what was the difference, guys? Can you tell me the difference which is over here with respect to simple or recurrent? Difference कहां पर आ रहा है यहाँ पे? Specifically,
बेस्ट जो
Tester.
आइडेंटिफाई किया उसका दोबारा
Tester.
से सेल्फिंग जो हम करा रहे
again, one by one. शशांक,
सर बेस्ट जो आइडेंटिफाई किया उसका जो अगेन हम सेल्फिंग कर रहे हैं विद टेस्टर के हेल्प से और वो
right? This is basically you have with the help of tester which you have identified out of these 4. They go out if you have selected 5. This you are doing in the simple recurrent selection or not? The selection with the original population. This you are doing with the simple recurrent selection also, na? है ना? Understanding?
यस सर
I'm focusing for the non-agri part. Non-agri student should understand this clearly. देखो, simple recurrent selection में भी I have original population. This particular GCA के लिए भी I have original population. Now, in the simple recurrent selection, if there are 100 population, out of 100, I have got 10 good plants I have selected. Over here, I am doing the same thing. 10 I have selected. But for general combining ability, I am going for a tester too. Now, I am checking these 10 plants also. In simple, I was not checking these 10 plants. I was growing their individual plant progeny. Out of these 10 plants, with testing with the tester, I have got to know that 5 plants are having good combining ability over here. So out of these 10 plants, further, these 5 plants will be going ahead with good GCA. And that will be going ahead with the individual plant progeny, and then we'll be mixing the seeds of this and we'll be repeating the cycle. This is the core difference over here which you will be finding. With respect to specific combining ability, what will change, guys? Now you see the specific combining ability. What changes you will be finding in that? Tell me. अब इतना समझ में आ गया तो स्पेसिफिक भी आ ही जाना चाहिए.
Sir, instead of open pollinated variety, we'll choose inbreeding.
Right. So now we are not concerned about this. Tester will be changed over here. Everything is going to remain same. And tester, I am going to use over here the homozygous inbred line instead of an open pollinated variety. Because I have to identify the genotype that exhibit high specific combining ability with respect to this particular tester or with respect to the selected plants over here. So this selected plant is performing high with this particular tester. Suppose this is performing high. So out of this 10 selection, out of 100, I have selected simple 10, but now I am going ahead with the SCA. So what I will be doing it with the testing it with the cross this particular inbred line. And I have got to know that out of 10, these 5 are really performing well. Now these 5 will be going ahead based upon the testing which happened with respect to the inbred line. The only difference you will be finding over here. So let me show you over here. Tester used over here, the modification in RSGCA is nothing but the tester uses homozygous inbred line. The aim is to identify genotype that exhibit high SCA. So superior plants will be selected as we were doing in both the steps. Cross with the specific inbred line. Whatever will be best will be taking, and selfing will be happening of that particular out of 10. Suppose 5 were best, so selfing will be happening of these 5 plants over here. And their selfing, the seeds which we are getting out of selfing, they will be test crossed and their seeds will be mixed, and then further it will be utilized as the original population. The cycle will be done again. Nothing, this is SCA. Again over here, the advantages of GCA is we will get to know additive gene action, helps in synthetics and inbred also, maintains genetic variability. Okay, this is over here, this helps us in understanding the non-additive gene functions. Then there is one reciprocal recurrent selection. What happens in reciprocal recurrent selection, guys? What happens in reciprocal recurrent selection? Tell me. बताओ भाई क्या होता है?
फिर इसमें 2 हेटेरोजाइगस टेस्टर का हम लोग यूज करते हैं.
Right. How we utilize this?
जब दोनों चीजें इंप्रूव करनी हो, GC और SC.
देखो, right. Over here, you will be finding 2 population. Now you will be taking 2 population over here, guys. 2 parent population. Suppose this is 1 parent population over here. And this is over here, the second parent population. Okay, so over here, what you will be doing, you have to select. पहले स्टेज में आप क्या करने वाले हो? यहाँ पर फर्स्ट ईयर में क्या करने वाले हो? ये जो आपने ग्रो करा है, what you will be doing over here? You will be selecting, suppose out of these 100 plants or out of these 10 plants, you have selected 5 superior plants of A. So what you will be doing now when you have selected this with 5? What you will be doing next? Tell me. Over here, you try to understand in reciprocal recurrent selection, first year will be this. Second year, you have to make the test crosses over here. This will be test crosses. Okay, so how this is happening? This is also original population of A. This is also original population of B. For A, B will become the tester. For B, A will become the tester. So suppose out of these 100, I have selected 10. So this 10 will now I will be testing it with the B population. So so what is becoming? B is becoming as a tester for A. And out of when I have done this testing over here, I have found that my 5 plants were good over here. So I have selected these 5 plants over here out of this test cross. So this is the test cross of A. I have got to know about 5 over here. In B, what you will be finding when you are again selecting the 10 plants of B, suppose. Now for this 10 plants of B, this A will be acting as the tester and you will be growing it over here. And then you will be evaluating it and selecting it. That out of these 10, my 5 plants of B are giving me superior result. So this you can identify it as A cross B. This you can write it as B cross A. I will be growing and evaluating the test cross progeny and I will be identifying the superior test crosses. So यहाँ से जो 5 मिले हैं मेरे को over here, I have got these 5 plants. I will be again selecting, I will be going and seeing these 5 plants over here and doing the selfing of these 5 plants only. So in the next year, in the field, what will be grown? The selfing of these 5 plants which are selected on the basis of this tester, which is being done with the help of B. Whereas over here, which is being with the help of A, similar thing you will be finding over here. Okay, let me show you the write-up also, you will be understanding in a better way. And then the same things will be repeated over here. See, selected multiple superior plants from population of A and B. Self each plant, also make test crosses A and B. In the examination, you show like this A, B. Then in the second year, you will be showing the test crosses. Okay, over here, test crosses you will be showing. Now you will be evaluating the test process and you will be finding that out of 10, 5 were selected. So you will be doing the selfing of 5. So you will be going in the third year like this and growing these 5 plants over here. So you can show like this. The third year, I will be growing the selfing of the selected plants. So यहाँ पर लिखोगे आप. And this is written over here too. In the year third, grow selfed seeds from parents that produce superior test crosses in the year 2. So year 2 is for test cross progenies, which all are not utilizing it. It's just to understand us that out of these 10 plants, these 5 were good. Intermate selected self lines within A, within B, free अपना selfing करेंगे. And then again we will be getting a mixed seed population over here of A and B, and the cycle will be repeating again. So repeat cycle for further improvement over here. Okay, so remember this particular part, like this you will be making or writing in the examination. So this is particular part you will be finding. See the advantages over here. Again, simultaneous improvement of GCA and SCA also, which is required for synthetic and composite varieties also. This is your recurrent selection, guys. Directly in the examination, how question will be coming. First, they will be asking you about the difference. Second, they might ask you about the schematic diagram or the procedure of recurrent selection. Third type of question they might give you on the basis of merits and demerits of this particular recurrent selection. So 2-part question will always be there. Might be a 10 marker, might be a 20 marker. Okay, so this is one part in recurrent selection over here. Now let us understand backcross method, guys. Backcross method, let us understand. These are difficult ones, that's why I have picked it first, backcross method, so that we can invest our energy over here, like backcross method. Why do you do this backcross methods? First of all, what is the need of this backcross method? Tell me, what is the objective?
सर स्पेसिफिक
In
कैरेक्टर्स
common,
को ट्रांसफर करने के लिए.
ठीक है. That over here, anybody else? Satakshi, you were saying something.
in the common variety, अगर कोई एक भी character, if single character is undesirable, तो सिर्फ उसको change करने के लिए. Rest everything is, the traits are good.
Right. देखो, suppose I have a variety over here which I am utilizing, which we are utilizing for past 10 years, the wheat variety. Now what you will be finding over here, that this wheat variety is the best variety which we are having in our country. But in recent times, there is one disease susceptibility which is coming in this particular variety. So I want 99% of the traits of this particular wheat variety, but I want one trait to get added. That is the trait which I am having in my wild variety of this wheat. I don't need a wild variety, because wild variety 99% things are not good for, favorable for us. But that 1% part is that it is having the it is having the disease resisting gene over here. So that gene I want into this particular part. Then it will become for me 100% favorable. For this, I utilize the backcross method. So to transfer a gene from donor, basically why? Because there might be something resistant, gene resistant or disease resistant trait might be there in the wild variety, which I need from here. Because of that, we utilize this backcross method. Now the problem comes over here that this particular gene which I want from this wild variety or any variety for the sake, land races suppose, it might be in the dominant gene, it might be present as a dominant gene, or it might be that trait must be present as the recessive trait. This can happen. For example, over here, suppose disease resistant trait is there, and in that particular variety, disease resistant trait is shown by capital R, and this is showing me disease resistant quality, that it is resistant to certain disease. Or rust you can take. It is resistant to rust. R is good for rust, so we will be taking R rust. So it might be present in dominant form. Or it might happen that this particular trait express itself when it is in a recessive form. And here it becomes a bit problematic for us to understand backcross in recessive part. So over here, my rust resistant feature is shown when it is present in recessive trait in this particular wild variety. So that is how the backcross methods change when the required gene isn't present in dominant form or the required gene is present in recessive form. So two types of stages will become, or two types of forms you will be, it will become, and it will be specified in your question also if they will be asking you. If not, then you will be showing these two methods under backcross. Okay, so let us understand the dominance backcross one, dominant phase of backcross. Non-agree must understand this much will be more than enough. Non-agree students, I'm telling you. Okay, and try to understand. If you will be having any doubt, try to ask too. Okay, over here, now I am going ahead with the backcross methods over here. Okay, the backcross will be needing, there will be a terminology which you will be utilizing for backcross methods. So what will be the terminology, guys? There will be one parent and then will be another parent. Suppose this is my original parent over here. So I want the traits, 99% of the traits of my original parent. So what I can call this parent as, suppose this is E, suppose this is B. So I can,
Recurrent parent.
so I'll be calling this parent as a recurrent parent. ठीक है, terminology ध्यान देना. And I'll be calling this P parent as, because I want only one trait or two trait from here. So I'll be calling it as donor parent. This will be my donor parent. Now I am concerned about one gene over here which is present in the form of capital R, capital R. So this is my dominant trait transfer which I am doing. So that means this is not present in this form in this particular parent recurrent part only. So that's why it is present in smaller. That is showing that this is susceptible to rust. This is a rust susceptible. This is a rust resistant. Okay, the first you have taken these varieties over here. So what you will be doing? You will be doing the cross of these. Crossing will be happening over here. And when a crossing will be, selective crossing will be happening over here. So what you will be getting over here, guys? What this particular generation will be known as? F1. F1 will be coming over here. In F1, what will be the progenies? The genotypes of progenies will be in F1. Again, Mendel law. Put Mendel law over here. For the non-agree, I'm telling you. If I am doing this hybrid combination of small r, capital R, what will be the genotypes I'll be getting in the F1 generation? F1 is my first generation, progeny generation. So over here, you will be finding that just make the Punnett square and you will be getting capital R, small r, capital R, small r, capital R, small r, capital R, small r. That means you will be getting only one type of genotype. That's why I'm writing capital R, small r over here. Is this gene resistant or gene susceptible? The progenies which I am getting over here, all, suppose 10 plants I have grown over here, all are gene resistant or susceptible? Tell me, guys.
Resistant, but 50% of the character from other parent also.
Right.
That we did not want.
देखो resistant तो है, but the problem over here, why we are going ahead with the backcross is because over here resistant trait which we wanted is there, but now 50% are unwanted trait of this donor, because 50% gamete are coming off this R which is needed and 50% of gamete are coming up of this capital R. The small 50% we need, but this large like capital R 50% we don't need. So that's why we have to go ahead with this back crossing. Means now we have to increase the content of the parent over here. So that's why now what will be happening? My RR will be going ahead with the back cross one. So over here you will be finding that back cross one will be starting, guys. This will be known as back cross one. Why? Because now this hybrid F1 will be crossed with the recurrent parent. So that means small r will be crossed over here. Okay, this cross will be happening. Now, what will be the progenies which will be coming out of it? What will be the genotype of the progenies which will be coming out? Again, if you don't know, you simply put the Punnett square. Put one gametes at one side, one parent gamete at one side. You will be getting that, okay, these type of gametes you are getting over here. That means two type of genotype you are getting over here. That is capital R, small r, small r, small r. So in your field, you will be finding some plants will be having the disease resistant trait and some plants, sorry, rust resistant trait, and some plants will not be resistant to rust. So what you will be doing over here, you will be selecting the rust resistant plant and you will be rejecting the rust susceptible plant in this field. Automatically you will be knowing this. So we have to retain only the capital R, small r. This is BC1. But over here now, content of small r which we needed, how much is the content of small r which we have in this particular backcross one? देखो यहाँ से फिर से you are getting 100% of capital small r. Over here you are getting 50% of small r. So when gametes will be formed, so 50% small r will be there. And when over here gametes will be forming, 50% small r will be there. Right? Sorry, 25% small r will be there. ठीक है, तो total percentage in this of small r will be 75%. And you have to go till 99%. Try to understand. That's why you will be saying that my exact copy of my parent I have got. Also one gene is added, that is of rust susceptibility or rust resistant characteristics. So that means you have to again do the back crossing. This percentage will increase. Again do the back crossing. So at least for 3-4 generation, you will be going ahead with the back crosses. At least BC1, BC2, BC3 till BC7 you will be going, and then you will be getting 99% of this capital R, small r. And then you will be going ahead with this variety release. Okay, let me show you over here. See this backcross method. यहाँ से आप पकड़ो इसको, transfer 1 or 2 desirable traits over here from donor parent into a superior but deficient recurrent parent over here. So what is the prerequisite? Superior recurrent parent, donor parent. Target trait must be highly heritable. Number of backcrosses 5 to 6, which is needed so that we have to recover the recurrent parent genotype, that is 98-99%. ठीक है, applications आप देख लेना. Applications are simple over here. Just let me show you the procedure over here. Parent A, genotype smaller because this is susceptible to rust. Capital R, resistant to rust. What you will be doing, like this, you will be showing it, guys, in a neat and clean manner. Small r, capital R, F1, that is resistant, but 50% of P parent is there. So we don't need that, right? So we will be doing the backcross one. Backcross 2 to BC6, like this you can write. Not every backcross you have to show. You can show that BC6 to BC7 similar performance will be done, and nearly all genes in the BC6 and BC7 stage will be 99% from the parent A. Then you will be going ahead with the selfing. And after that you will be going ahead with the selfing of that particular backcross now. Okay, and then you will be going ahead with the PYT and CYT, preliminary yield trial and preliminary yield trial for the local varieties, coordinated yield trial for different stations, agroclimatic stations. You will be going ahead, and then you will be doing the variety release over here. This is one thing. What if the trait is recessive? The problem comes over here. Now I am taking my trait as a recessive part. Okay, that means my over here the donor parent, that is B, is having this rust resistant trait as what? Recessive. This is recessive. That's why smaller. Earlier I was taking capital R. Understand the difference. And my recurrent parent over here is having the dominant trait over it, dominant susceptibility over here. Obviously, it will be inverse to this particular donor parent. Okay, that is what is shown by capital R. Now, what will be happening in this particular stage? Now you will be going ahead with the cross F1. Over here, you will be writing a recessive trait transfer. A recessive backcross you are going to understand. Now, when you will be doing the selfing, or when you will be doing the cross of this particular parent over here, you will be getting F1 as capital R, small r. Is this capital R, small r is resistant or susceptible, guys? Tell me.
Susceptible.
Susceptible. Everybody should understand why this capital R, small r is susceptible. Why this is susceptible over here? Non-agree
Since
student
it's
should understand. So tell me.
since it's determined by the recessive gene, so it cannot express itself right now.
Right. As per Mendelian dominance, law of dominance, you will be finding that the expression of the dominant allele will be there. And dominant allele, capital R, shows rust susceptibility. Rust susceptibility. And you know, if now resistant gene has to, if resistant gene has to express itself, it should be in a homozygous condition. Smaller condition. So that makes us understand that, okay, whatever I am getting in the field is not at all required for us because it is all rust susceptible. ये तो trait चाहिए ही नहीं हमें. So for this particular trait to come out, what we will be doing? We will be going for F2 generation. F2 generation means we will be doing the selfing of these particular traits over here so that the recessive trait should express. So capital R, small r, capital R, small r. When you will be doing this, again, those who are not able to remember what will come out, you simply do this particular Punnett square, capital R, capital R, capital R, small r, small r, capital R. This is same, small r, small r. So 3 types of genotype you are getting. Why I am not writing 4? Because this is same. Okay, so you are getting this capital R, capital R, capital R, small r, small r, small r over here. In this field, you are getting this particular part. Which of the genotype is favorable to us out of these 3 in this particular field? Which of the genotype is favorable for us?
Small r, small.
Smaller is favorable to us, right? Over here. Why? Because over here, this is susceptible, we will be neglecting this and we will be taking ahead this capital R, small r. Now, what we will be doing with respect to this? Suppose I have selected small r over here. But small r is having my over here the percentage of this B parent more over here. So now I'll be going ahead with the Backcross 1 F2. What I'll be doing, this small r, what I'll be doing? Doing the Backcross with the recurrent parent, that is capital R, and the procedure will be same again. Capital R, small r, then over here BC, over here this F1 will be generated, F2 will be generated, then BC2 will be coming, then BC3 will be coming, right? Let me show you. This is cross donor, this is small r, over here recurrent parent, now it is capital R. All are susceptible when we have grown the F1. That's why we are going ahead with F2. F2 is giving 3 genotypes, but the favorable one is small r because it is the only resistant one. So what I'll be doing now, I'll be recrossing it with the recurrent parent. That way it will be turned as BC1F1. Ye terminology important hai. Again over here, BC1F1 will be done because we have to increase the content of donor, increase the content of recurrent parent over here. Then again selfing will be done. Again this BC1F2 will be happening. Then BC2F1, BC2F2. BC3F1, BC3F2. Segregating lines BC. So यहाँ पर second generation का भी use हो रहा है, F2 का use हो रहा है यहाँ पर. Otherwise, in previous particular method, there was only one type of selfing we were doing, right? So this you will be repeating till BC7, and then ultimately you will be getting the homozygous resistant plants that resemble the recurrent plant over here phenotypically. ठीक है, simple. Then you will be understanding the PYTs and CYTs. आपके 2-3 साल last के 1 साल you will be keeping it for PYTs, and the last 2-3 years you will be keeping up for CYT, and last 1 year for you will be keeping for the variety release over here, right? So these things you have to understand with respect to this particular backcross methods. Rest, the positives and negatives you can read, they are easy to understand, guys. ठीक है, उसमें इतना नहीं है. Then you understand that there is 2 methods which are remaining, that is one pedigree and bulk. Every year you will be finding question on this line, either pedigree, bulk, recurrent, Backcross, pure line, ठीक है, इनमें क्वेश्चन रहेगा ही रहेगा, और 2025 में you will be finding the question on pedigree. Okay guys, so tell me the selection case in pedigree and the bulk part. How with the help of pedigree method you are doing this selection, and how with the bulk? First pedigree you discuss. What do we do in pedigree, guys? Tell me. बताओ भाई.
So crossing two different parents.
Right. What is the objective of pedigree, first of all?
To trace back the parent history, ancestry.
Right. We have to keep a record of the parent history also, and we are basically performing the cross between two parents where we need the combined desirable traits. ठीक है? And then we will be selecting the superior homozygous line of these two parents over here. ठीक है? This is what is involved in pedigree method. So what we will be doing over here, first of all, we will be going ahead with, in first year, what we will be going ahead with, guys? We will be taking two plants over here, sorry, two parents over here, P1, P2. And we will be generating the F1 generation over here, pedigree method. So we will keep the records of every plant progeny which has been come out of in this F1 generation, that what cross is made with the parent B crop. Okay, so this all record has to be maintained in a ledger. Right, and that I'll be showing you too, that how it will be maintained. Just take two examples from there and you have to write it in the examination also, that thing. Okay, so what you will be doing, you will be space planting this F1 over here, which you will be getting over here, right? The progeny of these two parent over here. Now we will be basically doing, we will be going for the F2 part over here, because what will be happening over here, the selfing of this particular parent will be happening over here, guys. So in F2, we will be going ahead with the selfing over here, and then also we'll be doing the selection of certain. Selection will start in F2 over here, after doing the selfing of over here, over these particular F1. तो यहाँ पर बहुत सारे आपको देखने को मिलेंगे population, and over here we will be selecting some plants over here of superior nature. The superior 100 to 500 plants will be selected. Let me show you pedigree method. Because this way you can remember it, so that you can show it to the examiner also, that how you will be maintaining the ledger. Crossing ledger you will be maintaining. Now, what is the cross number and what parent you have crossed it with. ठीक है? Just let me show you over here. So pedigree method, selecting individual plants from segregating generation. Segregating generation comes from F2. Okay, and F2 is also known as segregating generation. Raising progenies of selected plants, recording data across generation to trace the ancestry of each line. So you have to maintain the data over here too. Again, the objectives will be same. What we will be doing, F1 will be grown to confirm the cross. In F1, we will not be doing any selection. So year 1, parent A and parent B will be grown. Year 2, the selfing or the cross will be done of these two, and F1 will be generated. There over here, no selection will be done. Then F2s, when selfing will be happening over here, then we will be doing the selection in the segregating generation over here. यहाँ से selection शुरू होगा. ठीक है, तो F2 when it will be generated, then start individual plant selection based on desirable traits, and each selected plant is labeled and numbered. And then repeat this particular part, F3, F5, what we will be doing? We will be doing the progenies rows. Individual plant progenies rows will be selected. Suppose out of these 100 plant in F2 generation, I have selected 10. So 10 lines will be created over here, because these are 10 plants, and 10 plants progenies will be created over here. And we will be selecting now within these particular progenies that which is better. Continuously in F3, F4, F5, we will be doing this. And F6 onwards, what we will be doing, superior lines are bulked and evaluated, and then they will be sent to PYT and CYT. Over here. Now, if you want to, देखो merits, demerits, I'll be telling you, but over here, F1 generation, You have space planted the F1 seeds, F2 generation, you have done the selection over here. Harvest each selected plant individually to maintain pedigree. F3, begin evaluation of progenies over here. F4, what will be happening? That again, continue family level selection, similar thing will be happening over here. And ultimately in F5, F6 stage, you will be getting the homozygous situation, and then you can go for the PYT and CYT over here. Okay. Somewhere replicated yield trial happen, somewhere it does not happen. PYT, CYT will be going, and then ultimately you will be getting by this pedigree method, you will be getting the desired results. What is the merit of over here? Maximum use of breeder skill. Best for simply inherited traits. Easy identification of transgressive segregants. The segregants which are superior, that outperform both parents, is transgressive segregants. And precise inheritance tracking also can be done over here. Okay, demerits time consuming because over here you have to maintain the records. Breeder dependent success is there because you have to selection, you have to do the natural select, artificial selection. So that's why it will depend upon the breeder skill. Okay, so there are chances of human errors. Early selection may be ineffective over here because the traits you would want will be coming in F2 and F3 generation. So this is over here the pedigree method you will be finding. The core difference in pedigree and bulk. Bulk method में what you will be finding and what difference you will be seeing with respect to the pedigree. इनके बीच में difference भी आ सकता है. Tell me the bulk method, guys. What will be happening in the bulk method? Tell me, guys, what will be happening in bulk method?
Sir, no selection up to 6 to 270 or
Right. This is the only difference which you will be finding over here. That means if we are not doing selection till 6th, 7th year, what we are doing then? Why we are doing this? What is the objective of this bulk method then? Because of this method, what is the objective you are understanding? What a plant breeder is trying to do?
To achieve the homozygosity.
One is homozygosity, but homozygosity, I am getting it by the pedigree method also. Over here, the primary objective is to let the nature do its work. Over here, natural selection, on the basis of natural selection, it is going ahead. We will be creating some epiphytic condition, means we will be creating some stressful condition while all these years, which will be surviving automatically. It will be one of the good, better variety amongst this population. That is with the bulk population you are finding. Other similar objectives will be same as of the pedigree also, barring we will not be keeping the records of the entire progeny over here. Okay, so bulk method में what we are doing. So over here I have two parents over here. I will be creating the hybrid. Now I was doing the selfing of this and creating this F2, right, in pedigree. And I was doing the selection in F2. But now I will not be doing the selection. I will be growing these individual plant progenies, harvesting their seeds, mixing it in bulk, and then growing the same seeds again in F3. Obviously, naturally, these particulars will be selected. So some plants will not be able to generate and will not be able to grow efficiently. So they will die out or they will be removed over here naturally, but we are not selecting it. Till 4-5 years, we will be doing the same thing. And ultimately, we will be getting this particular part in the end, the homozygous condition we will be getting in the end. And then basically, we will be going for the PYT and CYT. Okay, so bulk method, breeding method in which F2 and subsequent generation are grown and harvested in bulk. Selection is delayed until later generation. Procedure is same. F1 generation space planted. F2 to F6, 7 grown in bulk, harvested, planted next season in bulk. No artificial selection over here, unlike pedigree. F6, F7 onwards begin single plant selection over here. Then we will be doing the single plant selection over here, like we were doing in pedigree. But the delayed years will be of 6 to 7 years gap, we will be keeping it. And then in the later years, we'll be doing this yield trials, multi-location trials. Automatically, this one of the demerit of this particular bulk method will be, it will be time consuming. So time consuming will be there. No pedigree records. Labor intensive. Labor intensive, not in starting, but in later generation you will be finding. It is less labor intensive. This will be one of the merits you will be finding of bulk method, because in 4 to 5 years, you are not needing labor to basically identify which are the best varieties. And over here, you will be finding merit, simple, convenient, inexpensive, large population allow natural selection. Epiphytic conditions can remove weak genotypes. Epiphytic conditions are those conditions in which you are creating the stressful condition with respect to water, soil, fertilizers, light. Good chance for transgressive segregants and save time and labor over here too. Okay, these are certain bulk method you are finding over here. Now your two methods which are remaining is what are there? Pure line selection and mass selection. Pure line selection and mass selection. What do you understand by pure line selection? What is the objective of pure line selection, guys? बताओ भाई, प्योर लाइन सिलेक्शन में क्या मतलब? Objective of pure line selection. Tell me guys, what is the objective? It is the process of selecting the best homozygous. Are we introducing any new trait in the pure line selection part? No. In the mass selection, we will not be introducing any new trait. Whereas in your pedigree, in bulk, in backcross, in recurrent, we were utilizing new traits over here too, because two parents were involved. So one parent trait we needed over here also, over here also, over here also. Whereas pure line selection and mass selection are utilized to improve the already grown variety within its particular genetic constitution only. That over here, this variety's trait we have to improve, not by adding any other genetic feature, but the genetic over here which they are having, the genetic makeup which they are having, we have to improve that only. For that, we are utilizing pure line selection. So the purpose is selecting the best homozygous line over here or homozygous individual over here amongst the mixed population. For example, that can be a landrace, old variety, right? And then we have to form a new uniform stable variety over here, which should be genetically identical. Okay, so what we'll be doing over here, what is the procedure of pure line selection? In pure line selection, what will be the procedure, guys? How you will be doing this particular thing, pure line selection? Let me take you to this particular part only. देखो, pure line selection, again definition you will be getting. Utilize this PPT. PPT will be having better structured notes of BD Singh only. ठीक है? Pure line selection is the process of selecting the best individual, homozygous individual, and propagating its progeny to form a new uniform stable variety. All individuals in pure line are genetically identical. How you will be doing? It will be taking you 8 to 9 years. In the first year, you will be basically you are having this variety. In the first year, you will be selecting. Selection will start from the first year itself. So you will be selecting certain plants, superior plants, phenotypically, which are better than the others. And all these traits you will be seeing that, okay, these are the traits which are favorable to me. Then you will be growing these progenies. Suppose you are taking 10 plants over here. Then you will be growing these progenies over here, plant to row or head to row over here. Every plant progenies will be kept over here. Now you will be selecting over here also, eliminating, suppose this is eliminated, this is eliminated, this is eliminated over here. So weak, disease, segregating, poor performing progenies will be rejected, and over here the superior plants will be selected, and it will be happening again over here. It will be repeated for 1 or 2 years, then it will be going for the preliminary yield trial. That means these selected plants will be competing for the check variety of that particular area. Check variety means the local variety which is the best in that particular area. Okay. So that will be checked against these, and whatever progeny will be outperforming that particular check variety, we will be taking it, and we will be taking it to the 4th, 5th years where we'll be doing the coordinated yield trials. Coordinated yield trials will be including other research stations also. And whatever will be the best multi-location trials are nothing but CYT only. So they will be now selected, promising cultures are tested at multiple location, and those who will be having better traits will be selected, and ultimately they will be multiplied, named and submitted for the release. So what we are doing over here, have we added any new traits? No. We have just basically improved the already existing variety, already existing genetics of that particular variety. So results in genetically uniform, high yielding variety. Okay, required less time and genetic variability than hybridization. Now there are advantages and limitations which are written. Tell me about how it is different from mass selection. What difference you will be finding in mass selection when compared to pure line? क्या difference आने वाला है? बताओ भाई. Tell me guys, mass selection pure line में क्या difference है? Tell me. Basic difference what you are finding between pure line and mass selection. देखो, mass selection is one of the oldest method by which these varieties are being developed over here. Again, the selection will be on phenotype, but over here no progeny testing will be involved. What do you understand by this no progeny testing, guys? Anybody can explain this no progeny testing? बताओ भाई. Anybody?
Sir, no testing with local variety.
नहीं, that every time you will plant breeder has to test it with the local variety also, local variety, and have to do the PYT and CYT. This they are not talking about. That is what I want you to understand. Anybody else? देखो, what we were doing over here, there in progeny. Suppose this is 100 plants I have grown of the variety X which we want to improve. 100 plants are there. I have selected 10 plants which is superior in quality. Okay, then in the next, what I'll be doing in the pure line selection, I'll be glowing the progeny of these 10 plants like this. So every plant will be having certain seeds. Every seed I have grown into this row, this particular column I have grown like this. Okay, so this I was doing in pure line selection, making a separate progeny testing over here. And then later on, it was tested with the local variety. Now over here, I will not be doing this. What I'll be doing instead, I'll be selecting first similar way superior looking plants based on observable traits. I will be mixing the seeds. Suppose out of 100, I have selected 10. Similar thing I was doing in the pure line also. In pure line, I was separately growing them. But in mass selection, I will not be separating it. Out of 10 seeds, each plant I am getting, out of these 10 plants, from each plant I am getting suppose 10 seeds. Total seeds will be 100. I'll be mixing them and then growing them again over here. And then selecting it and letting them have this PYT with the check varieties. So that means check varieties are not mixed. Try to understand. Sometime in second year, sometime in third year you will be. And then we will be going for PYT. Subse simple as PYT, CYT will be going for. And ultimately in the 8th, 9th year, we will be releasing this particular variety over here, multiplied, named and officially released. Understanding the core difference over here is the individual plant progeny will not be there in the mass selection. In pure line, there will be. In pedigree, was there individual plant progeny? Pedigree method. Were we growing the individual plant progenies? Individual plant progenies. बताओ भाई, पेडिग्री में देख रहे थे नहीं देख रहे थे? Tell me anybody. अगर इसका आंसर ही नहीं दे पा रहे फिर तो दैट मीन्स यू आर अगेन गेटिंग कंफ्यूज्ड. देखो ओवर हेयर, think about the pedigree method again. Pedigree is about having the two different parents over here. I am getting F1 over here with these two parents. But in F1, I was doing no selection. I was going for F2. In F2, I was doing the selection. I have told you 10 plants. Then I was going for this individual plant progeny. These 10 plants will be going for individual plant progeny. Whereas in bulk, I was not directly doing the selection in F2. I was doing the selection in F6 and F7. Understand this. Was I growing this individual plant progeny in recurrent selection? क्या recurrent selection में grow कर रहा था? बिल्कुल grow कर रहा था, right? In every recurrent selection, you will be finding this individual plant progeny. ठीक है? Mass selection में you will not be finding it. So advantages now you can take, limitation you can take of these particular mass selection. In this way, you can complete this particular line. Now you try to solve the PYQs which have asked. If you find any problem with respect to the PYQs, you can connect with me. You will be finding all the answers in the PPT itself. The next part we will be trying to do, just wait, na? We'll be trying to do the rest of the topics over here. The important topics like heterosis. Okay, all these topics we will be doing the next part. But these next parts are simpler in nature, like 2 to 3 pager for these particular parts. Okay. Origin 1 will be your homework. Whatever I have taught you in the lecture will be more than enough. Okay, new lectures I am uploading that you must see. Okay, new lectures will be uploaded on your portal. There is some issue which is going on, but it will be uploaded soon. Okay, guys. So this much is enough for this particular session. In the next session, we'll be completing all other aspects of the plant breeding. If you have any doubt, you can ask. Otherwise, I am ending the video, guys. But if you have any doubt, you can ask. ठीक है फिर guys.
Sir, please send PDFs of both previous lecture and this lecture.
Previous का तो मैंने भेज दिया था, I guess. I have sent the previous one.
Sir, group पे भेज दीजिए ना sir.
Okay. I'll be sending it. ठीक है.
हाँ, okay sir.
Okay, thank you so much, guys. Chalo guys, let us start today's session. That is enrichment cum revision session part. Basically, this is the second session for paper number 2. And in the last part, we had started plant breeding. This is plant breeding's second part. Before proceeding ahead, just confirm me that you are like, am I audible to all of you? Just confirm me once, guys. Am I audible?
Yes, sir.
Yes, sir.
To everyone, right? Good, great. Sneha, great. Amalam. Let us start our class over here. We are going to cover the leftover topics of plant breeding. Vijay, I have seen your message. Try to speak. Priya also I have seen. Try to speak in the class, right? Try to be connected over here. Now, till now, like in the last part, we have seen certain parts over here which were very, very important, and we have seen that with respect to the syllabus. Plant breeding is one of the core component of paper 2, as I have already told you, right? Paper 2, plant breeding is utmost importance, just like CBG, cell biology and genetics. This we have covered holistically, and this we are going to cover today. We will be finishing this plant breeding part. One thing I'll be telling you, those who have those who are from FCE and have the access to test series also, after this, try to attempt the test, because these 3 months are left solely for your optional preparation. Optional and other means subject, like you can pick up ethics and few more subjects, guys. This should be the strategy. If you will not study wholeheartedly at this point of time, you are shortly going to fail in the mains if you will reach in the main stage, because this is the rank making months. Okay, November, December primarily. And those who are not able to clear prelims in their last 3 years or last 3 attempts, then January onwards, you shift to your specific prelims preparation, right? January onwards. But if you are able to clear prelims, then I would suggest you to go ahead in the month of Feb onwards. You can think of your prelims preparation specifically. Right, this should be the strategy. So strategize every move right now if you are serious about this preparation. Otherwise, as I have already told you, people get stuck in the mains itself in their 2-3 attempts. Why? Because scores fluctuate over here. Sometimes 710, sometimes 690, sometimes 720, and the cutoff will be 730. Try to understand this pain right now, otherwise you have to feel it, and you will not be having any more attempt to give and rectify, improve your mistakes. I hope you will be able to do that, guys. Okay, strategize, take my help if you want. Over here, we had covered most important topics in the last class. That is this particular part from where every year you will be finding the question. Heterosis, we are going to see this modes of reproduction we have seen. Okay, and rest of the things we are going to see in this class today. Now let us start basically by understanding this also heritability also I have told you in the last class. Okay, this heritability is written in your CBG. All right, so over here, let me now go ahead and let us discuss about this topic of origin, evolution, and domestication of crop plants. Then we are going to center of origin, law of homologous series, and this, because over here you will be understanding all other topics. These topics frequency is alternative, means 2025 they have asked, if 2024 they have asked question from here, then the next time they will be asking 2026 or 2027. Okay, try to understand in this way. So let us understand about the domestication first of all. What is this domestication? ठीक है, ये बहुत important चीज है for your understanding purpose. And as you are an agriculture optional student, then you should have a clear idea about it. What do you understand about this, guys? Domestication का मतलब क्या समझते हो आप? What do you understand by domestication? Tell me. What do you understand about domestication?
Domestication means when we make a plant adaptable to our local environment rather than wild environment.
Yes, what you want to say that we have basically trained a particular plant to work to adapt accordingly, according to our needs, right? This is domestication. We are growing it as per our condition, wherever we want, whenever we want. Not whenever basically, but in sync with the climate, right? This is domestication. Domestication comes from a Latin word known as domus. And domus means home. Try to listen carefully what I'm going to tell you now. It will be like, try to think whatever I'll be asking you over here. Now, domestication, we did of two things, early humans. Can you tell me exactly, or not exactly, but roughly when this domestication process started? If you are reading history correctly, you must be seeing that when this domestication process has been started of two things. What are the two things which we domesticated basically? Two species, I would want to say, that is plant, this kingdom, and this animals over here. Right. Now, exactly tell me if you are good with your history portion, ancient history, if you have read R.S. Sharma, old NCRT, new NCRT. Can you tell me when there is an evidence of domestication of plants?
General during the Mesolithic periods, 10,000 BC.
Great. Yeah, this is the rough understanding I need. Okay, 10,000 BC, somewhere around, you will be getting the evidences of that humans tried to domesticate the plants. Over here, they were domesticating these plants. That means a particular land must have been grown of a particular plant species which is not originally found over there. It might happen that the origin of that plant is in Central Asia, but it is grown in the Indus Valley in that particular time frame. So that means somewhere around these historians, these archaeologists got evidence of domestication of plants in around 10,000 BC. Whereas in the earliest evidence of domestication of animals, as per old NCRT, R.S. Sharma, can you tell me what is the time given over there?
Sir,
Now, this will not be asked anywhere, guys, but you have to in sync with the rough idea about when these things were happening. You cannot say that, जैसे one of you have replied IVC. IVC से तो पहले बहुत पहले हो चुका है. IVC is around 2300 to 750 BC. So we are talking about the domestication process has somewhere started between or roughly around 10,000 BC of plants as well as animals. That means we are utilizing plants as per our needs and animals as
Yes.
per our want.
Mesolithic, Neolithic क्या?
Now tell me, was this domestication process important, or how, like domestication? Was it important or is it important, this domestication process which had started? इसी को आप एक और नाम दे सकते हो, that this is what was going on is agriculture revolution. You try to understand in humankind or in mankind, ठीक है, this human species was one of the species among all other species which were existing over here. Can you name me that what are the other homo species which were existing parallelly to us? Like plants are having their cousin species. Like this, we were having our cousin species at that point of time. Can you tell me other species name? Like we are Homo sapiens. At this point of time, during like 20 lakh BC to 50,000 BC, somewhere
Sir,
around.
sir, we are Homo sapiens. There is Homo erectus,
Right.
Homo
Then we
habilis.
were having other species of human also, that is Homo erectus, Homo neanderthals, Homo habilis. That means on our planet Earth, there was a time when all these species were migrating from here and there, living their life on hunting and gathering. That means something has happened, which basically because of which you will be finding that Homo sapiens are the only human species which you will be finding on the earth. Every other species have been eradicated from here. You are just getting the fossils of Homo neanderthals, erectus and habilis. And Homo sapiens in this time frame was not at the top of food chain. Where were the Homo sapiens? अगर मैं आपको Homo sapiens के बारे में पूछूं, where were they with respect to this food chain? कौन सा लेवल आप आइडेंटिफाई करवाओगे इससे? Humans. And humans during, or this Homo sapiens during, I would say 20 lakh BC to 1 lakh BC. What do you think? Today humans are at the top or at the bottom? Today humans at the top or at the bottom?
Top.
We are at the top today, guys. We can control every other species which is existing on this earth. We can manipulate them, we can control them, we can put them into zoo, despite of the organism size and ferociousness. Doesn't matter. But humans are capable of this today. But if you will be seeing in this ancient history, prehistoric times, humans have been in the lower rung of the food chain. That means we were at the primary or secondary consumer over here, not the higher consumer. And we used to feed upon the dead remains of the carnivores over here. Then we used to feed upon their dead remains. After all the top carnivores will be feeding on them, then we used to wait and do this scavenging activity over there. So something has happened which took humans from this particular rung to the top of the rung. Right. And basically you will be finding that there are 3 revolutions. Now in different books, different revolution will be writing, but 3 revolutions have happened because of which you will be finding we are now becoming like everything is revolving around us because we are able to control everything around us. And we are known as, like, what are these 3 revolutions? Can you tell me? बताओ भाई, शुरुआत में कौन सा रेवोल्यूशन हुआ? One is cognitive revolution. Cognitive revolution over here. Cognitive revolution means we are able to use our brain capacity over here. With respect to any other species, we were. आमलाम थोड़ा अपना वो साउंड बंद कर लेना, म्यूट कर लेना, आवाज आ रही है, या फिर जिसका भी है उसका एक बार देख लेना. So we were able to utilize our brain capacity as compared to other beings in a much better way. We became erect, so our hands got free. That means our appetite got reduced. That means our cranial capacity increased. So one, this cognitive revolution was happening, and because of this, we could utilize the natural forces like fire and other thing. When we started utilizing this fire, when we started utilizing these plants, when we started utilizing these animals, we became one of the best species in the food chain, or at the top of the food chain you will be finding. And this happened rapidly. This happened, we were at over here and we became like we came over here, right? In the evolutionary process. So one is cognitive revolution. Another one is you will be finding agriculture revolution, which happened. The technology, that food which we are eating is nothing new. The same wheat you will be finding, wheat variety, this maize variety, rice variety has been found by our ancestors, domesticated during this time frame. We are going to discuss that. Now, cognitive, I will not be discussing, not the scope of this class, but you try to understand what I'm like when we see the history of this domestication of plants. Now, third revolution which you will be finding, that is the scientific revolution, which changed the entire scope of this world. And an example, if I would want to give you, that this scientific revolution was not done by basically the Indians or the Asians. The scientific revolution came because of the Europeans. In fact, Europeans are so ingrained in our everyday atmosphere, everyday schedule, is because of this scientific revolution. The language we are speaking, basically the language we are speaking, the culture we are in, the clothes we are wearing, somewhere we are affected by this scientific revolution, and it is something revolutionary in nature, which happened in the 15th, 16th century. Okay, so that also we are going to discuss, not the scope of this class. The scope of this class is agriculture. A revolution I'll be trying to tell you over here. So agriculture, evolution, these three revolutions are important for a human species, and we are concerned about this agriculture revolution. The first step for this agriculture revolution started when this domestication process was started by the Homo sapiens. Now, is this domestication process important, or was this a very, you can say, a significant step which is important for us? And if you are saying that yes, this is important, sir, then why do you think that this agriculture revolution or domestication process was really very beneficial for us? Tell me. The question, first of all, this. Unmute करके बताओ.
Yes, sir, it is important.
Yes, it was important. It is beneficial. Why do you think so, Sneha?
Sir, because human has some needs, so they want to segregate the superior quality of plant which give more yield for
Okay.
livelihood.
So you are saying that domestication was beneficial. Why? Because
Want to separate the wild variety from the suitable variety which human needs.
you want to say that they wanted to have the good variety amongst the wild variety, right? They want eatable variety, which you want to understand,
Yes,
right?
sir. Yes.
Okay. Going to
And
it,
sir, also,
just wait. Let me write the Sneha's point over here, that the better variety you want to say of a particular crop species. This is what you want to say. Okay, that this agriculture revolution basically gave us this, or domestication of cropped helped us in selection of the better variety of crop species. All right, next.
sir, first of all, it is the foundation of human civilization. So उसकी वजह से plus regular food supply ensure करने के लिए भी it is important.
This is one good point highlighted by Shashank. Relevant point, that is foundation of human civilization. But not this also, the second point which you have said is the regular food supply. A regular food supply. How this regular food supply will be helping? कैसे हेल्प करेगा रेगुलर फूड सप्लाई?
सर टू सस्टेन देम, क्योंकि ऐसा इम्पोर्टेंट नहीं कि हर टाइम उनको एनिमल अवेलेबल ही हो, to
Right.
hunting and gathering के लिए.
So what you want to say that regular food supply is needed in times of when there will be dearth of something, basically when there will be crisis of this food, right? So they want to have a sustainable food supply. Because of that, they wanted to go for this domestication. Any other thought you want to tell me, or any other thing you want to highlight over here?
So particularly during the Mesolithic period, that is 10,000 BC, when domestication starts, the aim is the human society, they are moving from one place to another place rapidly. So they have to carry some seeds so that they can domesticate another place to produce that same food for eating purposes. That is the main cause of domestication, right?
Say it again, Amlam, I could not understand you.
Sir, during 10,000 BC, when this domestication concept starts,
Okay.
at that time the main aim of the human society is to moving from one place to another place rapidly. So for searching of a particular food, if they stay there, they will not put get that food throughout the year.
Okay,
So
again, you want to say
that
that yes, they thought that it will be beneficial for them to grow the same variety in a particular place so that they don't move from one place to another in search of food, right?
yes, yes, sir.
This is your point. Okay, this is all the
So sir, create a society.
right. Any other point you guys want to highlight, guys, that this agriculture revolution was beneficial for us for this particular purpose?
So another point also, day by day the population increases. To feed them that
This is the point which I wanted to listen. That because of this one thing happened, that is population explosion. Population was increasing because of this agriculture revolution. Now to feed more population, you need to grow more. Right? This is also one point. Great. Now anybody else want to highlight any other point? Somebody messaged also, Priya. Enough food gave way to other profession to emerge. देखो, let me tell you now, let me give you a thought now, a perspective now. Okay, and the perspective which I am going to give you is by one of the greatest scholars you will be finding, and they have given this perspective. Let me tell you.
Point.
हाँ, बताओ भाई, तुम बताओ.
Another point also, at that time there is no irrigation facility. They have to grow the crops in the banks of rivers. So to feed the extensive population, the productivity should be increases. For this also maybe domestication be adopted at that time.
देखो, now let me tell you first of all this domestication process क्यों हुआ उसके बारे में बताते हैं अभी. लेकिन but this domestication process beneficial or not? Let me just give you a perspective now. Listen quietly. देखो, this domestication of agriculture was, as per some scholars, is not at all beneficial for humankind. It was just a miscalculation that we domesticated these plant species, and now we eat what our ancestors have domesticated during 10,000 BC onwards. How and why this is not beneficial? Let us take the argument one by one. The first argument which comes to our mind, and this is ingrained in our mind because we have not thought the other way, and also we have not given it the right amount of time to think about, right? So first argument is which I found it more relevant is that the regular food supply we will be having, and later on human civilization was the outcome of it. It was not, the early humans were not thinking about the civilization, it was the outcome of it, right? So one particular thing must have thought by our early people, by our ancestors, is to have a regular food supply. Why not must grow a particular wheat variety which is in the wild? Let us grow it year by year, and when we are growing it year by year, then we are selecting the best species. Automatically the best species is coming to our way. But as per some scholars, archaeologists, anthropologists, they argue that the early humans, which I'll be calling them as foragers, and the agriculture societies over here, you will be finding like this. Agriculture people over here, agriculturists, Let me give them the name. First of all, foragers and agriculturists, when they got transformed into this agricultural society, what happened was this agriculture revolution giving us economic security, which we were talking about. You will be finding that archaeologists, scientists have ignored or completely rejected this fact. Why? On two terms. First of all, nutrition wise, the agriculturist societies were way too weak and eating less nutritious food. Why? Because in their diets now, majority of the diet consist of these staple food, cereals, and cereals also contain two variety, barley and wheat. Try to understand it carefully. Whereas when they analyze the foragers' diet over here, their diet was diversified over here. Diversification of the diet was there, including meat, fruits, and these cereals which they were growing from the wild. So economic benefit, the nutrition aspect, you will be finding agriculturist, the initial primitive agricultural societies were not that much nutritious, were suffering from malnutrition. So foragers, first of all, you will be finding that their life was much better in terms of the food or in terms of the cuisine they were having, the variety of food that was on their table. So this is the first point which gets neglected over here or which gets rejected over here, you will be finding. The second point comes that agriculture revolution have not, what do you think? The agriculture revolution have made the life easy of the people as they are growing more? If that is the case, then why at the lower rung of the today's society there are peasants, if agriculture have given out an economic boom? Let me give you a second thought now. Dekho, foragers, what they used to have, they will be roaming around and there will be not a dearth of food. This is a wrong concept which we have been fed upon, that there might not have been a regular supply of food. They were not needing this regular supply of food at all, because at their disposal there were variety of food which was available, right? Second thing, the most of their time was going in, you will be finding hunting and gathering, right? They were not, and these agriculturist life, you will be finding most of their time when they shifted from this hunting and gathering to agriculture, what you were doing most of the time during the day, they were toiling this field. They were understanding the basics of how to grow a particular crop. And when they understood, first of all, that wherever the wheat seeds are getting fallen, their wheat plants are growing up. So they understood that seeds are important. So what they started doing? They started taking these seeds in a particular land. For land, they used to clear this land by you must be knowing that particular what type of farming we say, that slash and burn farming. This is a primitive agriculture practice which were utilized by our ancestors, right? They used to clear this field. Why they are clearing this field? So that they can grow one type of eatable crop species over here. That is, for example, of this wheat species we are growing. What they will be doing over here? They will be basically removing the weed so that there should be no competition with respect to nutrition, space, light. What they will be doing over here, they will be basically taking the water and giving irrigation over here, bringing this water over here in this field. Sooner or later, like, it is not a single step from where you will be finding, it is an evolutionary step over here. So what was happening in this point of time, they also understood that when seeds are dug down, then they give better yield over here. Then the practice of hoeing, practice of this plowing started, right? So all this, they were spending much of their time in the fields at that point of time. And what happened because of that? This point is right, that we started getting food supply. This we are getting over here. Though this food supply is not at all nutritious. So because of this, two things happened. One, population began expanding. Population explosion started happening. Why? Because now the women are staying when they are, because 4 or 5 months they started to place their household over here. But later on, you will be finding that when certain varieties they started to grow, and when they become more familiar with this crop practicing, they started to have permanent villages now. So when they started to have permanent villages, now the women were getting time to raise children. Earlier, these foragers were not having that much time to raise the children. That's why population was under control naturally. Whereas over here, you will be finding population explosion happened. People multiplied. And when people multiplied, it is not connoting that they were having a good life. It is not signifying that. People were having a miserable life than the ancestors, than the foragers which we were having. Why? Because they have got the traces of malnutrition, of infant deaths in huge quantities. In huge amount, you will be finding that this malnutrition was there. Infanticides were happening at that point of time because of less food or malnutrition was there. So this trap started to happen, guys. As much as food supply we were having, our population was also increasing. So ultimately, you will be finding that throughout history, the peasants have always been on the lower rung. Despite you will be finding that till 1950s, we were primarily an agricultural society. तो होना तो ये चाहिए था, producer should be at the top rung. But this never happened. Rather, they were being utilized by the elites of the society. So domestication per se is not at all beneficial for the humankind. It was just a miscalculation, which we cannot now redo over here because all of the population is further dependent upon this particular revolution. And this is the way we think that we are dependent upon, or we are meant to live in this way only. Because there has been this evolutionary process from 10,000 to the present 2025. Right, so domestication perspective, which I am trying to give you, is one of this particular perspective, right? Second, what you can say about this domestication is then why domestication is considered, why domestication is considered successful. Domestication is considered successful because only you will be finding that there are humans, the human DNAs or humans basically multiplied because of this. We are not saying that the ratio of survival of this human species was higher or lower, but we are saying that among all other species, human species multiplied significantly over here because of this domestication process. We are not taking into consideration the happiness of the people. Just like we take the GDP and we don't take our happiness over here into count. Economy, if I tell you that a particular company is performing well or not, you will just see its revenue. You will just see its profit basically. Just like in the evolutionary analysis, when you will doing of any species evolutionary, this analysis, if you will do, you will make that particular species a success or a failure based upon their DNA copies. And humans were able to multiply rapidly over here because of that. Just because of that, it is considered to be a success to a particular species. But when you will be seeing it from a point of view of animals, these early humans started to domesticate animals too. And they started domesticating animals for certain purposes. One for food, second for transportation, third for their agriculture activities, and for other purposes also these animals were started getting domesticated. And these animals primarily was, you will be finding they were not very ferocious. These animals were completely docile, which can be captured, which can be controlled. So you will be finding over here pig, horses, cows, cattle, sheep. Like these animals you will be finding they were domesticated over here. And they were domesticated in the most cruel manner, if you will see. Again, I am telling you, early people were not at all human-centric. Okay? Human-centric means that I am thinking about from my perspective only. Early people were not so. Individualistic mindset was not there. This human-centric vision is a modern idea that we will be thinking about from an individual perspective. You try to understand, and you will be understanding it from the religion perspective also, though this is also not the scope of this class, so I'm not dealing with that too. But human-centric means that early people used to think about that in this particular earth, we are just one species. The other species is having the same relevance. That's why they used to worship the tree. That's why they used to worship the water where they are getting the river. That's why they used to worship the sun, because they think that they are vital over here and they are holding the same importance as we are holding. That's why the religion, the faith emerged at that point of time was animistic, where they will be worshipping the people tree, where they will be worshipping the bull too, because that is giving you, that is the fertile, and somewhere the kettles are giving milk because of this. Slowly and slowly, what started to happen, when we are becoming one of the powerful species, human became human centric. Human centric means now they are only thinking that every other thing which are placed in this universe or in earth primarily is or are for them, are for these species. That everything which is existing is for the benefit of this one species only, human. That's why the deterioration started. That's why the religion got changed from polytheistic to monotheistic. Now in the world you will be finding the major religion which is existing is a monotheistic in nature. All right, now this is a broader perspective about domestication, how we are doing domestication, like was it beneficial or not. This is just a broader perspective. This will not be asked anywhere. Now how the question comes over here, we are going to understand that. Okay. So history of plant breeding, you will be finding that over here you come, that plant breeding is nothing but selecting the best variety. So in ancient time also we used to do it by selection of crop varieties. In ancient texts like Kautilya's Arthashastra, and over here you will be finding the references to plant propagation and hybridization ideas too. Okay, so over here, ancient India, we are having certain things. 1900 to 1950, we will be finding the first hybridization program, the plant breeding programs you will be finding in the modern way. In 1905, it started in Coimbatore. 1912, IRI was established in Pusa, and that is Bihar you will be finding. Later on, get transferred from here to other way. Okay. Early breeders developed improved varieties of wheat, rice, cotton, sugarcane through selection and hybridization. Nothing to remember from here too. Later on, you will be finding that after independence, there was huge push which was given to agriculture research. And their development of semi-dwarf high-yielding varieties happened. Green revolution happened because of M.S. Swaminathan, B.P. Pal, C. Subramaniam. Right. And in 1970, you will be finding the ICAR also established the All India Coordinated Improvement Project. You will be finding over here. Okay, objective क्या है plant breeding का, as it is higher yield, improved quality. This you all know, guys. These are self-explanatory in nature. Just give it a read and you will be able to write it. There is no doubt about it. Okay. The thing which we have to go over here is just wait. This concept of center of origin, first of all, you try to understand. The center of origin, directly question comes in the examination from this line. Center of origin. If you will be finding that how these crops spread, can you tell me how this basically wheat variety crops all over which are present in the world, were they independent of each other or they are connected, like these have spread from a particular place of of African continent or Middle East? Can you tell me that how these crops have been spread? बताओ भाई. Tell me.
So
How these crops have spread? Yes, please say Sneha.
through domestication.
Through domestication, but what I am asking that how these crops, for example, the varieties which were growing over here, the varieties which were grown over here, there are center of origins, right? As Vavilov have given 8 centers of origin. Were these origins Independent in nature, independent means the people who are growing these things in that particular place was not knowing that in Africa there was something that is grown, was not knowing that this Iran that there was something that was grown. They were not knowing that in India wheat barley is being grown. They were not knowing that in China that was this plantation of this tea and all that was happening at that point of time, or rice was grown in China at that point of time. So were they independent of each other, or the theory is like they spread from a particular origin, like this is particular say Middle East, and the wheat spread from Middle East to other areas. So there are two ways of understanding it. What do you think? What is the acceptable theory over here? बताओ भाई.
Acceptable theory given by what's the concept of origin of theory? N.I. Pavlov.
So what is the accepted theory? Were they independent of each other, or they were basically transferred from a particular region to other areas? They spread from one particular area to other areas.
So they spread from one particular area to other area.
This was the common, this is what I want to highlight. This was the common misconception amongst the students. Let me clear that. You think that this agriculture revolution or agriculture crop species was also spreading like the way the humans were spreading. It is said that humans began spreading from this East Africa to other continents. But this was not the case with agriculture.
Means that wild varieties are present, but domestication started from that particular area.
Right. Just listen now. So over here, you will be finding that people were migrating, humans were migrating from East Africa. This is the most acceptable theory. But with respect to agriculture, when they started settling over here, they were independently growing what they used to grow over here. These Middle Eastern people were growing wheat as they used to grow wheat. They were not knowing about that these people in South America are growing maize. These South American people were not knowing that China people are growing rice. That's how this theory center of origin emerged. The origin of thinking of N.I. Pavlov, if you will find, it got from here only. That they started thinking about that over here he gave that this is the center of origin theory. Because certain crop plants were emerging at this point as this particular place independent of other areas. That's how he initially gave 8 center of origins. Later on added 2, 3, 4 something he has added later on too. But 8 center of origin, we should remember that, okay, these are the places which he gave in his theory. Now you understand, again, I was taught you in the last class also that these revolution or these theories are being put up by Europeans. Pavlov, Darwin, any other anthropologists, archaeologists, historians you choose, the geneticists you choose are from here. Mendel you will be seeing at this point of time Europe. So Europe was becoming a major hub at that point of time of this scientific revolution, which I was talking about. So ये आप थोड़ा connect करके रखो, because the agriculture knowledge which you are using can be utilized in your essays also. ये essay में भी आपका highlight करता है. Every knowledge which you are gaining from these GS subject or optional subject can be utilized in essays, right? So try to utilize all these things, guys. ठीक है, so center of origin. Cultivated plants not distributed uniformly. So कौन से पहले 8 center of origin हम देख लेते हैं. Just wait, it got stuck. कौन से 8 center of origin है? Tell me.
China.
One is China. China is one center of origin you will be finding. And for the examination point of view, try to remember 2 to 3 crops. 2 to 3 crops more than enough for a particular center of origin, right? तो कुछ तो देखने की कोशिश करना आप. तो यहाँ पर अगर मैं just wait, it got stuck, I guess. Yeah, so over here you will be getting one China. Tell me.
Hindustan.
Second is Hindustan, India.
Asia.
Third,
Asia.
Central Asia. Asia तो पूरा ही
Central Asia and Asia Minor.
Then Asia Minor comes in the Turkey region, Iran region. You will be finding Asia Minor. Then
Mediterranean.
America, Central America.
Mediterranean over here. And in
Central
Africa, you are going to find Africa के को क्या बोलते हैं अफ्रीका वाले को?
Abyssinian.
Abyssinian.
South America. South American उसको बोलते हैं.
Abyssinian कहाँ पर पड़ता है एग्जैक्टली अगर आप मेरे को बताना चाहो तो?
इथियोपिया और एरिट्रिया.
इथियोपिया. Abyssinia. ठीक है. This is Abyssinian plateau. Asked in your prelims examination where is this Abyssinian plateau? This is fifth. Sorry, this is sixth. Now seventh.
Central America, South America.
Central America and South America you are going to find. ठीक है. So the center of origin. Try to remember two crops over here. Who proposed this concept? N.I. Vavilov. Centers of origin based upon diversity and evolution of crops. He gave that there are two center of origin. One is a primary center of origin. Primary center of origin is the crop where it was originally domesticated. Secondary center of origin is the crop where it did not originated but has developed high diversity due to adaptation and evolution. It might be because of migration, natural selection, or human activity too. Vavilov's contribution were that because of his contribution, IPI was established in Leningrad, and he proposed 8 major centers over here. And because of him, he gave this law of homologous series in a variation. Very important concept, though very simple concept. ये word से इतना simple नहीं लग रहा है, लेकिन है समझना बहुत ही simple इसको. Can you tell me now, first of all, this is are the 8 center of origin. China center, one of the oldest over here. What you can remember is soybean, and over here radish if you want to remember. Center you remember this rice, arhar, chickpea you can remember, right? कुछ overlaps भी आपको देखने को मिलेंगे. It doesn't matter. ठीक है. Central Asia में सबसे important जो आपको मिलता है that is wheat. Wheat was primarily grown in Central Asia. इसका origin ये है. Later on, it got domesticated or transferred to other regions. You will be finding that in America, there was very least trait of wheat. But now there is whole Great Plains filled up of these prairies जिसे बोलते हैं हम. वो सबसे ज़्यादा wheat वहाँ पर grow किया जाता है. Right? So wheat you can remember, linseed, peat, any 2-3 crops you remember so that you can write it in the examination. Asia Minor, you will be finding Turkey, Iran, like this you will be writing, and 2 species, like 9 species of wheat, alfalfa is grown over there. Mediterranean Sea, you will find the lentils, clovers, onion, garlic, you can write. Abyssinia, barley is important over there, sorghum, bajra, these type of crops you can remember. Central Asia में आपको ध्यान रखना है maize, because ये वहाँ की variety है maize वगैरह, ठीक है. And South America, you are having maize, potato, peanut, pineapple, any 2 varieties you can remember again. Don't try to give more like focus over here because कुछ भी लिखोगे तो भी marks मिल जाएगा. It is not about the crops over here. It is about the centers over here. You will be getting the marks because of the centers which you are writing, if they will be giving you this. Later on, certain modification was done by Vavilov in 1935. South America center, they got split over here. USA center also got admitted. Right, so all these things were certain improvement on his theory of center of origin. Now, what is this law of homologous series and variation? What do you understand this, guys? ये बताओ मुझे. Law of homologous series.
So characters occur in one species also found in another species, जो related species होंगे.
Again, Shashank, you are saying it right. Please say it again.
सर कैरेक्टर्स जो किसी एक स्पीसीज में मिलते हैं वो उसी के रिलेटेड स्पीसीज में भी फाउंड होते हैं.
Okay, what you want to say that, okay, right. Correctly said that what do you, this law of homologous series of variation. Law of homologous series of variation was given by N.I. Vavilov in 1920. And as per him, he wants to say that this law is analogous to the periodic law. Periodic law in the chemistry. In chemistry also you will be finding the groups, the elements which are present near to each other will be having the same properties over here. The similar thing he wanted to say. that these particular homologous series of variation means the plant species which is found nearby to that particular species will be genetically close. Why? Because similar variation is likely to appear in related species as well. So if I want a particular species, if I want a particular trait in a species which is not inherently present in it, I can go to it nearby or nearby variety, like nearby genetically same variety, and I can take the trait from that through plant breeding. That's why this law of homologous series of variation became important. Now, introduced by N.I. Vavilov in 1920, the law is analogous to the periodic law in chemistry. This statement says that species or genera that are genetically close, they exhibit parallel or homologous variation. Homologous same variation. If a certain trait appears in one species, a similar variation is likely to appear in related species as well. This is what they want to mean over here, right? And now the example, if you want to see, suppose a disease resistance is found in one variety of wheat. A closely related cereal like barley or oats will be having also the similar variation. Right, so wheat over here, barley example is there. So parallel variation makes it easier for plant breeders to search for desired traits in a related crops. So it is utilized primarily by the plant breeders, guys. Okay, so this you must understand over here. Utility, predictive breeding, we can do exploration, transfer of traits, expands breeding base over here can happen because of this. All right. Over here you will be understanding. Now एक बार आप domestication का देखो, this from here question comes very rarely, but domestication can happen by natural selection or by artificial selection over here. Natural selection, how that climate, biological factors will be working over here, controlling the population of a particular species. But whereas artificial selection is carried out by man according to their needs and uses. Primarily, artificial selection has been done by the people, early humans, because of which most of the varieties which we are eating today, either of wheat, maize, rice, any variety you take, 95% of the variety which we are eating are being domesticated by our ancestors during the 10,000 BC era. There is nothing much new which we have created which has become the part of our staple diet. And that's why it is highlighted to have a balanced diet, because cereal-based diet is leading to malnutrition. That's why India is one such country you will be finding, and you will be reading it in unit number 7 also of paper 2, that India is having this particular problem, one of the major country with this malnutrition problem, guys. That's why we have to launch one program with respect to this, that is Poshan Abhiyan and all. Understanding, because that has been in our diet. Because of this, you will be finding this core problem. Okay, now origin of domestication, you can read, nobody will ask you this, but you just have to give it a read. So 10,000, 8,000, no need to get into the specifics of it. Even if you will be writing 12,000 chalega, 8,000 chalega, but you cannot write 1 lakh BC around. This is absurd, right? So over here, these things you will be finding. Okay. Natural selection, artificial selection, what is the difference? Though this will not come, but once you just see, because I have added this in your PDF now. Just wait. One thing also we have to discuss is the introduction part. Introduction. Plant introduction. Plant introduction. Plant introduction is of two. Basically, what is this? The deliberate transfer of plant varieties from their place of cultivation. Now, this is given in BD Singh also and written in your syllabus too. So one time we are just saying it. Questions rarely comes from here. Again, I am saying. Questions comes on CEO. Question comes on germplasm, conservation. Only these two questions frequently comes. Otherwise, not everything which is we are reading right now comes in the examination, but you just have to give it a read. Plant introduction is nothing but a deliberate transfer of plant varieties to a new geographical region, but obvious. There are two types over here. One is primary introduction and the other one is secondary introduction. Primary introduction, when an introduced plant variety is well suited to the new environment and is directly released for commercial cultivation without any genetic alteration, without any plant breeding program. It is called primary introduction. What we have done, no modification, direct use. And it is less common in countries because the crops are not able to adapt to a particular surrounding, or we will be having advanced crop improvement programs that we will adapt a particular crop to our needs. Right. So example, you will be finding over here like some varieties like Lerma Rojo or Sonaro 64. Okay, these are certain varieties over here. Secondary introduction is but obvious, will be having that we will be first evaluating them. Then it will be going through certain selection and hybridization programs for its adaptability, productivity, or resistance which we need in a particular variety. That's why it is known as secondary introduction. It involves a modification of the original genotype, common in countries with strong crop breeding programs. An introduced variety is used as a parent or a base population for selection purpose. Right, again, Kalyan, Sona, Sonalika are certain example over here which you are going to find. Okay, one like in short, you can revise like this over here, or you can make the notes in just 2 lines over here, right? Now over here, you will be finding these are certain things which are given: the agents of planted introduction in historical times, invaders, settlers, traders, travelers. Just give it a read. This will never be asked. Now this becomes important for your prelims examination. Like the Muslim invaders introduced cherries and grapes which introduced from Afghanistan. Whereas the Portuguese introduced maize, groundnut, chilies asked already in PYQ. In 2025, again, there was similar PYQ in this nature. Right, so potato, sweet potato, guava, pineapple, papaya, cashew nut, tobacco. 4 things have already been asked by the prelims examination. In the prelims examination, you are going to find, right? So these are certain things you are seeing over here, guys. ठीक है, ये important रहेगा prelims के लिए. Let us move forward. By East India Company, you are going to find tea, lychee, loquat, Mediterranean cabbage, cauliflower. Just give it a read. It is a good slide over here for your prelims examination. Now, the institutional phase, you will be finding the major institutional journey started from 1946 and a major institution for plant introduction. Can you tell me, give me the name of major institution which handles this plant introduction right now? The major institution which is there is NBPGR, National Bureau of Plant Genetic Resources. How it came into vision, you can see it over here. 1946, first of all, Division of Botany, IRI was found over here. 56, second 5-year plan, Plant Introduction and Exploration Organization set up. 61, independent division set up by IRI, Plant Introduction. And 76, finally, we have reached up to this major institutional setup, that is NBPGR. Headquarter IRI, New Delhi. It involves in introduction, quarantine, evolution, maintenance, and distribution of the germplasm which we have brought for agriculture or horticulture crops over here. There are certain NBPGR substation also like in Shimla, Jodhpur, Kanyakumari, Akola, or Shillong. You can remember this, right? NBPGR pe question ban sakta hai anytime. This is important over here. Now, other agencies which are involved in plant introduction are FRIs. Now, these do complementary works or sometimes supplementary works also with respect to NBPGR. What are the core activities of NBPGR? Import germplasm, conduct exploration, inspect and quarantine. Just read it once and you will be able to remember in the examination. Nothing unique in this. Test, multiply, maintain germplasm over here. Distribute the germplasm on the request of scientists, universities, maintain detailed records, exchange germplasm, publish catalogs and newsletters over here, establish natural gene sanctuaries, and undertake improvement of special crops also over here. Right, so these are certain activities you are going to find with respect to NBPGR. This is the conclusion for your NBPGR. It acts as the node, central node for all plant introduction activities in India. All introduction, whether by government agencies, research institution, or individuals, must go through NBPGR. Right. So this is one thing over here. Just wait. Now let me tell you the purpose of plant introduction. This also might get asked in the examination. Till now that has not been asked though. What do you think? What is the purpose of plant introduction, guys? क्या पर्पस है ये तो आप भी बता दोगे. ये these are generic in nature. ठीक है, everybody can write it without specifically reading it too. Tell me, what are the purpose for plant introduction?
So introduce new crop varieties, high yielding varieties.
Right, high yielding varieties, new varieties if we want, we can introduce by this plant introduction methods. For example, maize, potato, tomato.
Here the disease resistant variety.
राइट, एज ए पॉइंट्स आने चाहिए एग्जाम में. डिजीज एंड पेस्ट रेजिस्टेंट हमेशा. प्लांट ब्रीडिंग के लिए कुछ पॉइंट फिक्स कर लो. यील्ड इंप्रूवमेंट, इंप्रूवमेंट ऑफ अ वैरायटी, न्यू ट्रेड्स, डिजीज, पेस्ट रेजिस्टेंट, और बताओ.
Establish new industries.
ठीक है, राइट. साइंटिफिक रिसर्च इफ यू विल बी डूइंग, देन न्यू these agriculture industries can flourish. Apart from this,
Aesthetic value.
aesthetic and ornamental value you can remember, because plant introduction happens because of this also. Lantana camera, all these weed species, all these which have become weed, was brought up in India for this aesthetic value only. Alright, और कुछ?
Conservation.
Conservation purpose also you will be getting. So same things you will be finding. Introduction of entirely new crop plant, new varieties, crop improvement, disease and pest which you have said, scientific research which you have said, aesthetic and ornamental value. If question comes, you will be writing in the similar way in the examination. One point example, if you know, right? Keep the format in this way, guys. Take a simple thing, this we have discussed over here. Now next is center of origin also we have discussed. Next, let us discuss about the germplasms. This also come frequently in the examination, guys. Germplasm. What do you understand by germplasm? Tell me first this.
What do genetic material?
All hereditary materials.
Right. It refers to a genetic material that carries the hereditary information of the species. Of a particular species, I have all the information. This is nothing but my germplasm. And why it is being utilized, how it can be beneficial for us. How it can be beneficial for us, germplasm.
So for breeding purpose, research purpose, trait improvement.
Right, all these things again, that it can be like if you want to improve a particular variety, to build, to basically ingrain some new traits. With this germplasm help us. It act as a raw material for plant breeding. Disease and pest resistant genes we can get. And traits like drought tolerance, yield improvement can happen because of this germplasm over here. So if I take you, just wait. Also tell me that how you will be doing this germplasm conservation. What are the various ways of doing this germplasm conservation? Can you tell me?
Sir, seed banks, in vitro methods.
Right. So germplasm, you can write it by ex situ and in situ methods. Like this, you will be converting it. Ex situ, what we have? Just now you have said it. Seed gene banks. More.
Field gene banks.
Field gene banks you are going to find. In vitro conservation.
So please,
Botanical gardens.
Right. Cryopreservation also somebody has said that also can be utilized over here. What about the in situ?
So botanical gardens.
Botanical gardens, okay. Apart from this, on
इन
farm
सीटू में सर
conservation.
नेशनल पार्क्स हो गए,
Yes,
बायोडाइवर्सिटी पार्क्स हो गए.
protected areas, biosphere reserve, gene sanctuaries, national parks, which you have read, all this will come in this particular part only, right? So let me make you see this over here. Just wait. Germplasm is total hereditary material in species. You have understood. Collection is having the storage of this of varieties, land races, wild varieties. Importance क्या है? Raw material, disease, pest resistant traits like drought tolerance we can get. Threat is genetic erosion, means when entirely if this particular germplasm get wiped off, it means a particular species we have lost. So there is a threat of genetic erosion which can happen over here. Definition again of germplasm you will be seeing over here. How we can maintain it? Ex situ conservation, what we have discussed, it is written over here. In situ conservation, on farm conservation, and biosphere reserve, gene sanctuaries, and national park you are going to find. All right. So over here, like certain genetic resources which are used in rice breeding is like submergence tolerance, drought tolerance, salinity tolerance, blast resistance. Learn any 4 examples from here, guys. Any 4 examples you can learn and you utilize it in multiple areas. Multiple areas you try to utilize. ठीक है? ये बाकी एग्जांपल्स हैं. इतने एग्जांपल्स में आपको जाने की जरूरत नहीं है, बट एक बार देख लेना that I have added over here some other examples also. What are the problems in germplasm maintenance? The problems you will be finding: the loss of seed viability might happen, high cost, risk of mislabeling or genetic change might happen if a particular worker over here is like careless about handling of this particular germplasm. Storage issues might happen. A vulnerability problem have seen with respect to the maintenance of germplasm, right? Okay. So solution is like cold storages should be built with long-term conservation methods. This has been done in USA, Philippines, and other areas of NBPGR repositories too. ठीक है, so ये आपको देखने को मिलेगा. Gene sanctuary is nothing but in situ conservation method where a protected natural area in center of diversity where crop species and their wild relative grows undisturbed. So the regions where these are grown is a northeastern region. You will be finding the Musa variety, the citrus, Oryza, Saccharum. You will be finding these are grown over there, right? देखो, one question has been asked that what do you understand by gene pool concept? Again, it is nothing but the same concept. The total genetic viability, diversity. Just wait, na. I hope my voice is clear. The gene pool refers to the total genetic diversity available within a species or a related species over here. This was coined by Harlan and Tevot. Yeah, as it is, learn kar lena, because this question has been asked already in the examination. The gene pool concept classifies plant genetic resources based on their degree of crossability. Classification कैसे कर सकते हो? Primary gene pool. It will be including the cultivated species and those wild forms or land races that can freely cross with it to produce fertile offsprings, like cultivated wheat and wild wheat. Secondary gene pool is including that species that can cross with the cultivated crops, but the resulting hybrids may be partially fertile or infertile also. This might happen. Third, tertiary is including these distant wild varieties, which can produce primarily these trial hybrids or no hybrids at all. And it is being utilized for a particular trait by using advanced techniques like tissue culture, protoplast fusion, or gene transfer over here. What do you understand by genetic erosion? Again, simple, self-explanatory, refers to the loss of genetic diversity within a crop species, especially the disappearance of traditional land races, wild relatives, and primitive varieties. Again, simpler in understanding.
Causes क्या है? That we have replacement of land races which has happened because of high yielding modern varieties. Urbanization, habitat destruction might happen. Monoculture farming practices, climate change and natural disasters also can lead to this particular problem. ठीक है, तो ये कुछ आपको देखने को मिलेंगे, कुछ प्रॉब्लम्स यहाँ पर. Now, if I ask you what is the importance of crop resource genetic conservation or crop genetic resource conservation and utilization, as it is, this is the syllabus line. What is the importance of crop genetic resource conservation and utilization? What do you think? What is the importance, guys? Again, same examples will be there.
Sir, food and nutritional security.
Right. Other point.
Sir,
For
prevents genetic
future use.
vulnerability.
ठीक है, बोलो और. बोलो क्यों चाहिए तुम्हें ये
Sir,
crop
for future use.
for future use?
Sir, to protect ITK, मतलब traditional knowledge.
Yeah, right. Safeguarding traditional varieties basically for future use, you want to say, or indigenous varieties we have to save, right? Climate change adaptation में help करेगा, genetic diversity plant breeding में help करेगा. So genetic diversity for breeding, safeguarding traditional varieties over here. Climate change adaptation, it will be helping you. Scientific research and buffer against genetic erosion. What you are understanding over here? देखो ये वाला टॉपिक हमने ओवर करा है जो आपका उससे रिलेटेड है, जो सेकंड लाइन है आपके सिलेबस की, ठीक है. What you are understanding, the only important thing in plant breeding is that particular part of selection and वो जो हमने देखा था वहां पर डिफरेंट सिलेक्शन मेथड, वहां पर थोड़ा सा आपको पढ़ने की जरूरत है. बाकी जगह सेम इंफॉर्मेशन यू कैन यूटिलाइज अकॉर्डिंगली, राइट. इसमें अब नेक्स्ट हम समझने की कोशिश करते हैं. जर्मप्लाज्म वगैरह तो हमने देख लिए. हेट्रोसिस हमने लास्ट क्लास में डिस्कस किया था. हेट्रोसिस आई गेस वी हैव नॉट डिस्कस, और हमने डिस्कस किया था. चलो देख लेंगे हेट्रोसिस को भी. इंपॉर्टेंट टॉपिक रहता है हेट्रोसिस एंड इट्स एक्सप्लॉइटेशन. वी विल बी सीइंग इट टू. देन वी विल बी सीइंग सोमैटिक हाइब्रिडाइजेशन. Over here, this particular part we will be finding, or we will be reading it in unit number 6, plant protection. Then these 2 points are related to genetic modification, guys. Okay, directly question comes that what is the benefit of this genetic modification, genetic engineering? Direct question comes over here. और एक ये molecular markers and their application in plant improvement, इसको भी हम. Now these are the topics of 2-3 pages only. ठीक है, these are not broad topics over here. So let us discuss one by one over here. If we will go in the understanding of, just wait now. जेनेटिक मॉडिफिकेशन आप समझने की कोशिश करो आप पहले. जस्ट वेट. Genetic modification. Genetic modification, what do you understand by genetic modification? Though you must be knowing it. Can you tell me what do you understand by genetic modification, guys? क्या समझते हो genetic modification से?
Insertion and deletion of what?
A particular
gene
you want to see, right? ठीक है, अगर हम बात करें बीटी कॉटन की, which is the only sanctioned crop over here which can be grown in India legally, BT cotton. Though other crops are also grown for particularly breeding and scientific purposes.
ठीक है, but not legally for large cultivation. BT cotton is acceptable one, has got the approval basically. So BT cotton is one major genetic modified crop you will be finding. What do you understand by BT cotton, guys? How this genetic modification has done in the cotton plant? Tell me. And why it has done? This is a cotton plant. Now cotton plant is being fed, basically is getting impacted by the pest. And
Cotton
this
bollworm.
pest is basically cotton bollworm.
Bollworm.
Sometimes these are pink bollworm, red bollworm, and other type of species you will be finding of bollworms. Because these are the pests which feed upon this particular cotton and the yield gets deteriorated over here. So what scientists have done, that they have found out one particular bacteria which can kill this particular cotton bollworm. And this they have found in the soil only. From the soil they have taken out this bacteria. What is the name of this bacteria? अब ये ऑप्शनल के लिए important नहीं है.
Bacillus thuringiensis.
Right, this is not important for your optional. This is more important from your prelims point of view. और जब आप वो पढ़ोगे science and technology, biotechnology when you will get it. Bacteria is a Bacillus thuringiensis. Just check the spelling once from the PPT. Don't worry. This bacteria, why this bacteria is unique, guys, with respect to cotton bollworm? Tell me why this particular bacteria is important with respect to cotton bollworm. चलो मैं बताता हूँ आपको. In Bacillus thuringiensis, this is a bacteria. This bacteria is having one gene. And the gene name is Cry1AC. And this Cry1AC produces this Cry protein basically. And this Cry protein basically, when is feeded by this cotton bollworm pest, it basically kills this particular pest over here, guys. That means it dysfunction the functioning of this particular pest. So when scientists got to know about that this cotton bollworm can be killed by this particular protein which is released by this bacteria, they thought why not insert this particular gene which produce this Cry1AC protein to this cotton species. And when there is transfer of one gene from another species to the another species, it is nothing but transgenic modification. Transgenic means two different species are there involved. So one is cotton species, one is over here bacteria. And then they have come up with the new varieties when they merge it with cotton, that is BT cotton. Now the BT cotton is going through multiple stages, guys. There has been first stage of BT cotton where Cry1AC was utilized. Second stage, Cry2AB you will be finding. And then Cry1AC also came over. Sorry, some other name is there with respect to the protein. And it is said that this third variety of BT cotton is basically herbicide tolerant too. Not only pest tolerant, but herbicide tolerant. Right, this is one thing about BT cotton. This I wanted to discuss. One very famous, asked in your prelims also, is a GM mustard. And this is a bit complicated over here, guys. देखो रोल वगैरह तो आपको लिख ही लोगे, वो मैं एक बार डिस्कस कर ही लूंगा. I'll be discussing that what is the advantages or role of biotechnology with respect to genetic modification. That you can write. But the functioning also you try to understand. What is this GM mustard? And how and why this GM mustard was created? Tell me now. What was the objective of creating this GM mustard? Who has created this? Can you tell me? This you should know, guys. This is, let me tell you, has been asked in your prelims also.
So ये जो the professor की ओर बोली होती है
कोई बात नहीं. The
दीपक
professor, you will be
दीपक
finding that Delhi ex-vice chancellor, Delhi universities,
दीपक पेंटाल
Deepak Pental and his team worked upon this mustard variety basically and did some modification which came to known as GM mustard. Also, other name is known as
धारा mustard.
hybrid
TMH11.
mustard 11. Now, the first question which should come to our mind. देखो, I am teaching in such a way that if a person.
Sir, DMX.
हाँ, रुक जाओ एक second. I am teaching in such a way that if a person is new to this preparation, they should know that what is this GM mustard. Now you try to understand. First of all, I am having this mustard variety. सरसों की variety है मेरे पास mustard variety. Now, first of all, this mustard variety is multiplied by cross pollination or self pollination. This mustard variety is a bisexual flower or a monosexual flower. Bisexual flower means it is containing both a male part also and female reproductive part also. So it always go into selfing most of the time, guys. So you know when selfing is happening of a particular species, will its yield will be better than the crossing or worse than the crossing? Inferior than the crossing you will be finding. So this Deepak Pental and his team, what they wanted, that they wanted to increase the yield and performance of this mustard. For this, they wanted to go ahead with this particular technique of crossing. This is the primary objective you try to understand. That this mustard should go crossing, because of which the hybrid which you will be getting in the next generation, which be having hybrid vigor, nothing but heterosis. And this I think we have seen in the last class too. Last enrichment class, heterosis मेरे को याद आ गया, शायद हमने पढ़ा. Or else in the lecture also I have explained it really well. So this was the primary objective, to increase the overall performance of mustard. Now the complex procedure over here is the way they introduce certain things over here which were all over in news back in when they were doing this experiment. And time and again, whenever there will be some article with respect to this GM mustard, these particular genes will be talked about more frequently. Now tell me how and what he did over here with respect to modify this mustard. Batao bhai. First of all, we will be taking two varieties now. Because he don't want this mustard to go for self-pollination. He want cross-pollination. So, but obvious, I'll be taking two varieties. I'll be taking over here two varieties. One, he has taken a Varuna variety. That is the indigenous variety which he wanted to grow over here. इसी का इसको yield चाहिए. The second variety which he took was the European variety, that is early Hera variety. Understanding? Now what he did, guys, tell me. अब क्या करेगा वो?
Transgenic line heterosis.
हाँ?
So heterosis breeding क्या?
Heterosis?
Breeding.
सर साइटोप्लाज्मिक मेल स्टेरिलिटी किया था.
देखो बहुत यहाँ पर देखो, बिल्कुल सही जा रहे हो दोनों. ठीक है. Amlam was saying that we should cross them, right? This is what you want to say, ना?
Yes, sir.
Now the problem here, बहुत अच्छी बात बोली कि यहाँ पर क्रॉस कर देंगे, जो हमें F1 मिलेगा वो हाइब्रिड ही तो मिलेगा. बट द प्रॉब्लम हियर इज, फर्स्ट ऑफ ऑल प्रॉब्लम को समझो. एवरीबडी अंडरस्टैंड द प्रॉब्लम. If we directly do their crossing, the problem here is that Varuna variety might go into selfing, because this particular variety is having what? Both male and female. This is also having but both male and female. What guarantee you are getting that F1 generation is a hybrid? There might be a chance that you are getting nothing but a variety of early Hera, or the progeny of early Hera, or a progeny of specifically Varuna. So your whole objective is defeated over here. You will not be able to get the F1 required F1 species. The whole amount of money you have put in investment or in this research has been basically gone into vain. right? So what is the right method now? Now you will think that how I will make this particular, which Shashank has rightly said, that how I will make this particular Varuna variety, because I want this variety to go into crossing. I'm not concerned about this. I definitely want that every variety of my Varuna should go and like cross with the other variety of early Hera over here. So what should I do over here? I should do something with the pollen which is getting formed. Male part को मैंने sterile बनाना. That's why he has said male sterile. I have to make this particular crop. That there should be no pollen formation, or there should be dysfunctioning of this pollen formation, which should happen over here. Now this can happen by two ways. You can say, sir, how I can do this? कैसे कर सकते हो ये pollen को निकालना? कैसे कर सकते हो? How you can do this? One is emasculation process, guys. Emasculation. That is hand emasculation. But is this possible that 1 hectare land you will be doing this hand emasculation with utmost accuracy? This is not a feasible process.
Insertion of zinc.
Right. So now he came up with different idea. And the idea was the genetic idea. In this genetic idea, you think that how far we have proceeded, that now we can transfer gene from one species to another species. How far we have come in with respect to our technology, guys. From stone technology, we used to have 1,000 years or 2,000 years before, and now we are able to do this. Now, what he thought, he will be picking over here certain genes now. Try to understand it carefully, because a lot of people, even the people who are scientists over here with this particular subject, are not able to understand it clearly. Tell me now what he will be doing. क्या करेगा? He will be interested in one gene which can make this plant male sterile. This is the requirement. When you have understood the requirement, now it will be easier for you to understand the terminology for these particular things. Now the gene which he has utilized is very famous and might be known by you all also. What is that particular gene, guys?
Sir, Barnes or Barster?
Master.
देखो, this is what I am saying right now. I am just talking about that particular gene which will be important for this, which will be responsible for this male sterileness, which I want in early this Varuna variety.
उसके लिए सर बास्टर और बार्नेस यूज होता है.
देखो, try to understand over here. Barnes, remember it for your life. And a where
Barnes,
na
Baster.
is coming, you try to remember where na is coming. That means it is related to the sterile part. Right, it will be doing male sterility over here. So Barnes gene I'll be utilizing over here. Now will my problem solve of this particular selfing? Now selfing will not happen. 100% one thing will be happening, that in this field there might be that my crossing of this Varuna and early Hera. And there might be one more possibility. Can you tell me, when I am doing their selfing now, there might be one more possibility. The progenies might be of
हीरा.
everyone. The progenies might be of early Hera too. यहाँ selfing इसका तो मैंने नहीं करा ना, इसको तो नहीं रोका. तो कुछ progenies early Hera की भी तो हो सकती है. Over here you will be finding. So one Barnes gene I utilize over here. Now, to not have this problem, that the selection problem in my F1 category over here when I'll be selecting these genes, because I want only the Varuna hybrid variety. That means I am concerned about this only. So something needs to be done. और वो something क्या है? That let me tell you now. First of all, now over here, if I am giving this gene, Barnes gene, to my Varuna variety, and this Varuna variety, when will mate with early Hera variety, they will be producing, suppose this A variety over here, the hybrid over here of Varuna and early Hera. Will this be required by me or not? Will this be acceptable to me or not? Tell me. Think. I want everyone of you to think.
No.
It will not be required by me. Why?
Because from that side, some seeds are available, present there.
Sunya, again. Why this variety will be rejected over here? देखो, when we don't understand clearly, that's why we will rectify this bar star and bar niece, and we don't understand their use. Try to understand. Try to go into the depth of it. Once you will understand this, you will remember till your life. Why I will not accept this particular varieties which is coming over here. I
सर
will
बाद
not.
में रेप्लिकेशन नहीं होगा इनका.
Yes. Why? Because over here, because in these varieties, you will be finding still this particular trait is there. This gene is present, and this gene is of Barnese. That means whatever variety, whatever seeds will be produced, they will be sterile. So why I will or why farmers will purchase from me these sterile seeds? ये तो grow ही नहीं कर पाएंगे. So again, the whole purpose is defeated. Now to avoid this, Deepak Pental and his team will be doing what? Will be doing something that he will again want a gene. He will again want a gene, and he will again want a gene for why he will be wanting a gene? To restore the fertility of particular
mustard.
of mustard. That means वो pollen बनाने लग जाए. मतलब ये जो बार्नेस का काम था वो रिवर्स हो जाए यहाँ पर. वो इसलिए ये चाहता है. So इसके लिए कौन सा जीन इसने इस्तेमाल किया? बार स्टर नीर और बार स्टार भी बोल सकते हो, बार स्टर भी बोल सकते हो. तो जीन विच हैज बीन यूटिलाइज्ड अगेन ओवर हियर वाज बार स्टर. बार स्टर. What this gene will be doing, and this gene was interested in what this gene were inserted to Varuna or early Hera?
Early Hera.
What will happen if I insert this gene to Varuna?
So sir, again same problem will be.
Again same problem will be having there. Both will be neglecting it. Right. So that's why I have introduced the gene in early Hera. देख लो कितना complex कर रखा है. This is a simple process, but they have made it really very complex over here. Because this is the only way you can go ahead. These are not very easy things. These are easy things while I am saying it, while you are studying it. But actually, when we are doing it on the field, it is one of the major complex process, guys. So this barster I have introduced now in the early Hera variety. That means when these two will mate together over here, what species I will be getting will be male sterile or fertile? Fertile. That means this will be acceptable now, acceptable. But now the problem will be that how will I differentiate between these two species? The early Hera only one or the hybrid which I have got? Because I only want hybrid. So my two problems I have solved. Now the third problems I have to solve. For this, I will be utilizing one more gene. And the name of this gene is
Bar.
Where I will insert this gene?
F1 progeny में सर.
F1 पे बाद में नहीं डालूंगा मैं, इसको डालूंगा तो पहले ही बार जीन को. लेकिन where I will be inserting in the early Hera or in the this particular gene?
Sir,
Early Hera or Varuna?
I think Varuna.
देखो, first of all you understand what is the objective of this bar gene. What is the importance of bar gene? ये highlight difference कैसे highlight कर रहा है? What is the function of this bar gene?
I think production boost करने के लिए है ना, उनका yield increase करने के लिए.
देखो, bar gene is responsible for basically it will make one thing which this particular DMH is famous for. This is herbicide tolerant, na?
Yes, sir.
Though they wanted to increase the yield, but one more character it has, herbicide tolerant. Now this bar gene is responsible for giving a resistance to
active herbicide ingredient.
Weed or herbicide. What do you understand by non-selective? Means in a field, if I pour some herbicide, it will be non-selective. It will be killing all the species which is existing in this particular plant. This is non-selective. It will not select. And what we have, the example over here of non-selective herbicide. Glufosinate. So that means if I am adding early Hera, this Varuna variety with this bar gene, will it become resistant to glufosinate or susceptible to glufosinate?
Resistance.
Resistant to glufosinate. And why we are doing it? Now I have this field. Now you understand my Varuna variety. I have introduced Barnase and Bar gene to this. This is early Hera variety. I have introduced Bar, what was the Barstar gene over here? समझ रहे हो? Barnase, Bar gene, Barstar gene over here to earlier. Now whatever species crossing I have done, suppose I am getting this hybrid over here, and somewhere I am getting this self of early Hera only also. So what I'll be doing now in the field in order to get this particular only early or only this Varuna hybrid.
सर herbicide डाल करके
Now I will be pouring herbicide over here. And what do you think? You are going right now. What do you think? Which will remain in the particular field? These all will be dead
white
because
वाला.
they are not having bar. Only white one will remain. That means only the DMH11 hybrid will be remaining over here. The experiment is completed now because this variety seeds will be having higher yield and they are also herbicide tolerant. I hope you are understood now the importance of Barnase, Bar and Barstar. This thing is not still completed. Why I am saying this genetic modification? Because we are introducing a gene from a particular species to a particular species. Over here the two species are. Why genetic modification? यहाँ दो species कौन सी हैं?
वरुणा early है ना.
Varuna and early.
This is the problem. These are same species, guys. These are plant species. Then how I can call it genetic modification or transgenic modification? है ना? प्रॉब्लम है ना? इसको हम कैसे बोल पा रहे हैं कि ये ट्रांसजेनिक है? समझ पा रहे हो? The question should be this. These two are same varieties. You think about the gene. Where we are getting this gene from? Barstar, Barnase, Bar. Where we are getting from? जिसकी मैंने शुरुआत में बात की.
बैक्टीरिया है. Bacillus bacteria I think.
Yes. Because these all genes are coming from a particular bacteria. Now I am talking about this. And like deliberately, I did not say this bacteria in the starting. Everything is deliberate, guys, over here. Okay? This is not I have left it. Because in the last, you understand that this is the bacteria. And this bacteria name is, can you tell me, Bacillus something you will be finding? Amylo something you will be finding. If I check karna, and this I will be having in the PPT. Though it will not be required over here to remember the name, but if you can remember, you can remember it. Okay. So Bacillus, this bacteria will be having
Bacillus amelopubensis.
liquefaction, something like that you are having, right?
Bacillus amelopubensis.
Now understand this bacteria is having these three genes. Understand again that Barnase gene, Bar gene, and Barstar gene. And this is being introduced in this Varuna variety and early Hera variety. Ultimately, to get a variety which is hybrid and which is herbicide tolerant. I hope this topic is completed now. Okay, that is DMH mustard, DHM mustard. Okay. Now let us go to the application part. जब ये दोनों समझ लिया आपने और if you have understood these two, these are the most important thing. Is DHM mustard is like legally acceptable and grown in our country?
No sir.
No sir.
It has not been, right? So this is also, you can say this is the first edible GM crop. Edible I am saying. Whereas if I say that first GM crop will be BT cotton, but edible which we can eat also, this is GM mustard. And because of that, because there are a lot of concern with respect to genetic modification crops, that's why we are also not releasing these varieties. समझ गए? ये था कहानी. अब थोड़ा आओ exam oriented, like how they are giving questions, like application of genetic engineering for insect and herbicide resistance. Now you can simply give the example, guys. Insect resistance principle will be that BT crops you are having BT gene, cry gene over here from Bacillus thuringiensis, crytoxin. ये सब आप देखो एक बार, ठीक है? Already very easy to understand. Then there are certain genes which are making basically this pest resistant, these crops pest resistant. ठीक है? Similar goes for herbicide tolerant over here, and the examples are mustard, DMH mustard, which we have made, very important, which you can remember. ठीक है, तो glufosinate and glyphosate you will be finding over here. This you can remember. ठीक है, genetic modification crop, BT cotton, there are golden rice also, but I don't think so they will give you. The only two things which they might ask in the exam will be BT cotton and GM mustard. ठीक है, many of there are also which are there, but they will not be asked. Use of GM crops in insect pest management. ठीक है, same thing is written again and again. Management of disease it can do. Management of weeds we can do over here. ठीक है, तो overall benefits will be it can increase yield, farm income, reduce use of chemical pesticides and herbicides, better crop quality, environmental sustainability. Again, same thing you are going to find over. Role of biotech in hybrid and improved variety production in crops and concept of hybrid vigor already we have seen. Okay, again you will be seeing these same things again and again. This you will be doing on your own, guys. Just see the content once. These are simpler in nature. Next topic which I should discuss over here is basically with respect to molecular markers. Let us discuss the molecular marker concept over.
Sir, I have one question.
Yes, Shashank, please.
Sir, to highlight a specific DNA gene.
Right. Molecular markers you will be finding. देखो मार्कर्स, सबसे पहले अगर हम मार्कर्स जेनेटिक मार्कर्स की बात करें, genetic markers, how many type of genetic markers we are having?
The DNA based.
The DNA markers, pre-DNA markers, two types.
अच्छा, डीएनए मार्कर्स की बात कर रहा हूं मैं. जेनेटिक मार्कर्स में कितने टाइप के मार्कर्स आपको देखने को मिलते हैं?
डीएनए मार्कर्स, प्री डीएनए मार्कर्स. उसके बाद प्री डीएनए मार्कर्स 3 टाइप्स है, मॉर्फोलॉजिकल, बायोकेमिकल.
हाँ, बायोकेमिकल. सेकंड वन, व्हाट यू हैव सेड इट? Biochemical markers. One is morphological markers. Second is biochemical, which you have said. And the third is which we have to study in more detail because in our syllabus line that is written. That is molecular markers. These three markers are the genetic markers. Why genetic marker? What do you understand by the markers? Marker is nothing which can mark or which can highlight something. So in the DNA, there are certain portions which are highlighted by either by morphological, either by biochemical, or either by molecular level. So these three things you will be finding over here. What do you understand by a morphological marker? Morphological marker से क्या समझ में आया?
उनके मॉर्फोलॉजी को हाईलाइट करना, लाइक लेंथ, ब्रेड्थ, कुछ उनका वैसी क्वालिटी.
इसी को क्या बोल सकते हैं? दैट इज ऑब्जर्वेबल ट्रेट्स. Morphology is nothing but what we observe. So you can say what are observed, and what is observed where? In the DNA, and DNA is heritable. So observable heritable traits controlled by single or multiple genes that can be visually identified either by flower color, seed shape, leaf shape. This is a morphological marker. This is having certain observational traits. ठीक है, this is one marker you will be finding over here. Second is a biochemical. What do you understand by biochemical marker? Tell me about the biochemical marker.
I think so, जो इंटरनल केमिकल्स जो प्रेजेंट है उनमें कुछ चेंजेस या कुछ उनको हाईलाइट करना.
Right, that means marked by the biochemical thing. Biochemicals can contain your enzymes, and enzymes will be utilized over here. And the primary enzymes like utilized is isozymes, which we call them. No need to remember because the topic will be molecular part. Okay, so just see it over here. certain storage proteins will be there. So they will be utilized like albumins, globulins, gyardins, no need to learn these words, for varietal identification. These are not affected by environmental factors like morphological traits, and these can be act as the markers or genetic markers over here in the biochemical category. Again, one category you are having, no need to go into depth of it. The third
Yes,
one
sir.
is the molecular marker. What do you understand by the molecular marker now? मोलिकुलर मार्कर से क्या समझते हो?
सर स्पेसिफिक डीएनए सीक्वेंस या रीजन को जो कि वेरिएशन शो करते हैं उनको मार्क करना, आइडेंटिफाई, ट्रैक और एनालाइज करना.
ठीक है. That means over here molecular means that these are based upon the direct DNA sequence. My voice is getting repeated. Please turn off the mic. So it is the marker based upon that. This is basically what that there is certain sequence in the DNA which we can mark over there, such as certain nucleotide, certain substitutions which are happening over here. So these are of different types over here in a nuclei, in a this particular nucleotide chain, nothing but DNA. That is present in particular DNA. For example, VNTR you must have heard about. What is this VNTR?
So various nucleotandem repeat something.
Right. Variable number tandem repeats you are going to find in fingerprinting also. I'll be telling you this DNA fingerprinting. उसमें भी समझोगे, but these are nothing but your molecular marker. Short tandem repeats means certain parts of a DNA get repeated every time. These are nothing but act as a molecular marker. So there are different types of molecular marker which we have to go through also. That this is short tandem repeat markers which are shown. That there are AG80, there are 7 repeats over here. Repeats mean again and again this sequence is getting repeated. Okay, like this you are going to find. So types of markers, morphological, just read it out. Biochemical marker, again you will be reading it out. Molecular marker we will be opening up. In this you will be finding it is based upon the DNA sequence. And there are major types over here. Now, for UPSC examination, we will not be going in detail of every of this particular techniques of doing this molecular markers. These are all techniques over here which you are finding. But one hack you have to do if you don't want to go inside into the information of how this technique is utilized, you can remember certain techniques over here which you can throw in the answers and which I'll be telling you also. That hack you should know. That is RFLP, RAPD, AFLP, SSR, SNP, ISSR. ठीक है, एक एक करके देखेंगे. अभी इसको भूल जाओ. कुछ आपने सुना होगा, कुछ नया होगा. We are going ahead with only two examples over here. Two techniques we will be keeping it fixed. ठीक है, so this one thing you have to know. Now other thing now over here. That is molecular markers can also be differentiated between two markers category over here. Molecular markers. And molecular markers, how I can differentiate now? On the basis of? Tell me guys.
सर उनके डिस्टिंक्टनेस या उनके वर्केबिलिटी पे.
ठीक है, thinking it right. But इनके कुछ नाम है academically which you have to remember. One is dominant marker and the other one is codominant marker. What do you understand by dominant marker? Dominant marker cannot distinguish between homozygous dominant and heterozygous individual because both will produce the same pattern, same bad pattern. देखो ये कैसे पता चलता है हमें. Suppose this is one particular repeat I am having, ठीक है, of DNA. This is my parent A, this is my parent B. Now over here I am having certain genetic over here, certain genetic over here of them. Suppose A, suppose B. And when I am like laying them on electrophoresis, electricity when I have given them, तो इनके DNA कुछ अरेंज हो जाएंगे ऐसे करके. Suppose ये यहाँ पर अरेंज हुआ. Suppose this particular B arranged over here. When I'll be doing their hybrid, when I'll be going for their hybrid, don't understand in this way. Just I'm giving you an example. When I am making their hybrid, I'll be getting the same band as of up one particular parent. Suppose in the hybrid, I have got the band of my parent generation over here. So it is replicating this particular part. So by the hybrid, I am not able to know that this I have got basically from which parent, or this I am not able to get that is the genetic material contains of this parent also or not. Because why? Because one parent has been dominant over here and it is showing the same band as of one parent only. It might get over here capital A, capital small b. Just for understanding purpose, I am telling you like this. And this happens in the band formation type of way only. So when I'm saying dominant markers cannot distinguish between homozygous dominant and heterozygous dominant. For example, अगर यहाँ पर A भी होता तो भी you will not be able to distinguish that from this where this AA is coming from. This might be AA also. So distinguish is less over here. And the patterns which we use over here is RAPD. This you have to remember. Can you tell me the full form of RAPD? Random amplified polymorphic DNA. For the examination, those who are non-bio students, non-biotech students, I will not be asking you because I have many which you will be seeing later it on also. Don't try to remember it, the definition if you don't want to go. Nobody will ask you a full-fledged question on RAPT. The good part is this only of UPSC. ठीक है, तो यहाँ पर आपके कुछ shortcuts चल सकते हैं. Though if you want to know more, you can know more by searching on it, or if you want some content, I can give you. But this is not required. RAPD you will be getting over here. Then one is AFLP. AFLP. What is AFLP, guys? Amplified
fragment length polymorphism.
Polymorphism. Good. Okay. Amplified fragmented length polymorphism. What are these? These are nothing but the techniques of separating these markers over here. These all are techniques which are being utilized. So by this RAPD, AFLP, what I am doing, I am separating these dominant markers over here. Okay. Now the next thing is codominant marker. What do you understand by codominant marker? कोडोमिनेंट से इतना तो समझ में आ गया कि दोनों का कुछ ना कुछ आ रहा होगा यहाँ पर, राइट? So codominant markers can differentiate between homozygous and heterozygous genotypes because both alleles are expressed and detectable over here. And over here also we utilize certain techniques. The techniques which are utilized over here, you can remember SNP. What do you understand by SNP? Single nucleotide polymorphism. What do you understand by, again यहाँ पर आप RFLP भी use कर सकते हो, RFLP, or you can utilize over here SSR2. RFLP already I have told you. SSR is simple sequence repeat. Everything is given in the PPT. Don't worry, I'll be showing you. Simple sequence repeat also you will be getting over here. Okay, dominant markers. Dominant markers cannot distinguish between homozygous dominant and heterozygous individual. Both produce the same band. RAPD, AFLP, you can remember, leave ISSR. ठीक है, ये दोनों ध्यान में रखो. Band appears, presence of allele over here. No band, absence, AA. Advantage is simple and quick, require less DNA, no proper sequence information over here. Limitation is can't detect heterozygosity. Less informative for population studies. Codominant markers you are having can differentiate between homozygous and heterozygous because both alleles are over here by the name itself you will be knowing. Techniques which are used over here is RFLP, SSR, SNP. Remember any two, SSR and SNP you can remember. So working principle over here, single distinct band, two distinct bands of one each allele, and a single different band which you are going to see over here. Okay, advantage is it detects all genotypes correctly, useful in genetic mapping and marker assisted selection. This we will be discussing just right now. Marker assisted selection. What do you understand by that also? Now, what do you understand by this word which is written over here? And this is only written in your syllabus. What is marker assisted selection, guys? The definition for it. Give me the definition for it.
So to identify association between molecular marker or gene controlling to improve the plant breed.
ठीक है, देखो marker assisted selection is the selection which I am doing now with the help of these molecular markers, which is helping me to select desirable traits before the trait is expressed phenotypically. Phenotypically, when it is expressed, when it is grown in the field, that I am doing, that is selection only. But with the help of these molecular markers, I am utilizing it beforehand, before they are grown phenotypically. Right, so marker assisted selection is a modern plant breeding technique in which molecular markers, DNA sequence linked to specific genes, are used to select plants possessing desirable traits before the trait is expressed phenotypically. It enables indirect selection for traits that are difficult, expensive, or time consuming to evaluate. Right, so over here, this you will be finding. Example, you should remember, I will be giving you certain. Can you give me certain example of marker assisted selection?
सर मेज क्रॉप में एक विवेक करके कोई वैरायटी गई थी.
ठीक है, मेज में आपने ये तो सही बोला, लेकिन विवेक का आई एम नॉट इन्फॉर्म्ड अबाउट दिस, बट देयर इज वन थिंग विच इज हैपनिंग इन मेज, that these markers are used to improve two content, that is the lysine content and tryptophan content in maize. और इससे क्या बनाना चाह रहे हैं? क्वालिटी प्रोटीन मेज. नाउ देयर आर different names of these varieties which you can see over here. Any other variety which you know of? राइस में भी काफी काम हुआ है इसका.
सब बासमती, पूसा बासमती.
Great. Over here you will be finding one basmati species and the other one which is very famous is the Swarn sub-1 species you will be finding here. Right, Swarn sub-1 species. The second species which I will tell you over here is the Samba species. ठीक है, सब कुछ mentioned है. You don't have to do anything. Submergence tolerance in rice is being seen in Swarn sub-1 species. And the sub-1 gene conferring submergence tolerance was introgressed into the high-yielding rice variety, Swarna, using marker-assisted backcrossing. The maize variety which we were talking about is quality protein maize. The gene was utilized this. If you want to remember, you can remember two varieties to yaad karo for your MAS topic. Okay, nothing is wrong with that. Blast resistant and bacterial blight resistance, if you want to remember, you remember. The genes and the problem it cured. This you have to remember over here. Now, what are the applications of MAS in, suppose I am saying in recurrent selection? What is the application of mass in recurrent selection? Molecular assisted selection in recurrent selection, how it is being utilized?
जीनोटाइप को आइडेंटिफाई करने के लिए यूज होता है वो तब.
What we do in recurrent, we try to select a parent again and again, right? Recurrent selection mein what we were doing, we were selecting a parent again and again. We were doing a similar cycle, not a parent, but we were repeating the cycle again and again. And why we were repeating the cycle again and again? To get the higher or better variety over there. Selection can happen in a better way. How marker assisted will be helping us in recurrent selection is that now breeders can identify individuals carrying the desired alleles, and they will be intermating those alleles which are desired of those plants with the other species too, so that we can get the we can get the required favored alleles. Right, that is how they are reducing the time cycle of this recurrent selection, nothing else. जो cycles ज्यादा लग रही थी उनको कम कर देगा ये mass. ठीक है, let me show you over here. So in recurrent selection, it aims to accumulate favorable alleles for quantitative traits by repeated cycles. Now this repeated cycle is the problem over here. So now using these markers, breeders can identify individuals carrying desired alleles at multiple loci and intermit only those increasing the frequency of favorable alleles. What is the advantage? Increased selection accuracy for traits with low heritability or late expression, which you will be seeing. And this has been utilized in a maize for grain yield under stress and non-stress conditions. बहुत सारे programs are going on. One of the program is this. Try to understand, even if you are not able to remember something in the examination, you try to create one if you are not able to remember it, and it will happen with some questions in the examination. Okay, so this is one part you will be getting over here. Now, how this mass is being utilized in gene pyramiding? Gene pyramiding, how you will be utilizing it? What is gene pyramiding, first of all? जीन पिरामिडिंग होता क्या है? Anyone? देखो, gene pyramiding is nothing but it is a technique, it is a breeding technique that will be combining. pyramid क्या है? Like this, it will be going like this, है ना? So it will be combining multiple desirable traits from different parents, not from one parent or two parents. From multiple parents, which are the desired genes, they will be multiplying it, and it will be included in a single genotype, in a single parent, in a single offspring. Like I want a disease resistant, yield also, quality also, all this I want in a one particular species. This is nothing but in simple terms, gene pyramiding. And this gene pyramiding will help me, will be immensely successful by the utilization of marker assisted selection. So now how it will be utilized? Gene pyramiding involves combining two or more genes, controlling the same or different traits. So now mass will be helping me to track the position of each gene in a particular parent or in a particular species offspring using gene specific markers, even if the genes are phenotypically similar or masked. So in rice, pyramiding of these species were done for bacterial blight resistance. Remember what particular part you can remember, guys. These are nothing but genes. Or kuch genes to you have to remember only, right? And in wheat also you will be getting these genes which are rust resistant genes. So this is being done in mass, is being utilized in gene pyramiding also. Now, what is how mass is being utilized in quantitative trait loci? Introgression. What do you understand by this introgression? First of all, introgression का मतलब क्या हुआ? इसकी vocabulary का मतलब क्या हुआ? Introgression. ये पढ़ा तो होगा आपने, सुना तो होगा introgression. I guess in one of the lecture also I have taught you this introgression, or just in one statement I have told you this introgression. Anybody with this word introgression? Introgression, you try to understand, it is the transfer of genetic material of one species. Suppose a species is there and a genetic material I'll be transferring into the gene pool of another species over here by repeated backcrossing. So backcrossing, when I was teaching you in the class, I have utilized this word, that is introgression. Introgressive hybridization also we can say this. What I am doing nothing but purposely I am utilizing the gene or the genetic material of this and I am adding to it with a particular species over here. This is automatically also, it happens naturally, and it also happens in like where you will be selecting artificial also it can happen. So this QTL introgressing happens over here. QTL introgression is the process of transferring specific quantitative trait loci associated with complex traits from donor to recipient genotype. Over here, we can use MAS tightly linked to the QTLs. Breeders can select plants carrying targeted QTLs during backcrossing. and minimizing the donor genome contribution over here. The Pokali variety over here you will be finding in IR64 is utilized by SSR markers. ठीक है, तो पहले मार्कर असिस्टेड सिलेक्शन का SSR RAPD over here different, ये आप ध्यान में रखोगे तो you can utilize it over here too. Broad tolerance was done in your MEs also with respect to MAS. Now this was the question which has been asked in the examination as it is, and it was on a difficult side. Why difficult? Because these are not as it is given in your academic books or the books we are having. Neither I have taught you in my earlier courses. ठीक है, that's why I am adding it over here. एक बार बस देख लेना इनको. It might be asked in this. MAS से बहुत कम क्वेश्चन आए हैं. You will be finding the repetition cycle is 3-4 years. In 3-4 years, you will be finding one question. The good part is you are going to sit for 2026. Be free. Question might not come from MAS now for another 1 to 2 years. 2026, 2027. There might be chances that question will not come. Okay, so this is one thing with respect to marker associated selection. ठीक है, सो ये हमने मार्कर एसोसिएटेड सिलेक्शन देखा. One over here now, the application. You tell me, what are the applications of molecular markers in crop improvement? This also they can ask you, and they have asked you once. Application of molecular markers in crop improvement. Now you have known certain methods. I have told you 3-4 varieties also how you can do marker associated selection. Tell me, what are the application guys? क्या क्या applications आपको देखने को मिलते हैं?
Sir, identification of plant species.
ठीक है, identification of plant species will be done over here, right?
Linkage mapping alleles.
The most important thing is genetic mapping you will be doing over here. So genetic mapping you will be adding. Good.
The selection of desired traits.
The desired traits which you can do over here with respect to this marker assisted selection, right?
So it can accelerate backcross also.
Backcrossing, that means MABC, you can marker assisted backcrossing. आप उसमें भी डालो. अभी मैंने क्या क्या पढ़ाया आपके इसको एप्लीकेशन कहाँ पढ़ाए हैं? Gene this QTL introgression. This should be the vocabulary now. One more thing I have taught you right now with respect to Priya, research genetic basis, right Priya? एक और चीज अभी बोली थी ना मैंने आपको. What I have told you guys? Gene pyramiding. Pyramiding में you can utilize it. Right, and in one more thing, you can utilize these molecular markers over here. Genetic diversity analysis, construction of genetic maps, marker-assisted selections, marker-assisted backcrossing, which you have said, gene pyramiding you will be finding, gene selection, hybrid breeding you will be finding over here, quality trait improvement, biotic stress resistance. Example you should have over here with respect to this. Without example, things will not work in this particular question. Okay, so this is a mass over here. Certain questions like this you will be finding in the examination again. Okay, now there are certain other things mass we have discussed majorly. Like, और कुछ important हमारे पास जो somatic hybridization, let us discuss now one important thing. Somatic hybridization. Yeah, this we will be discussing now. Because this application of principle of plant breeding, this is nothing but this is the combination of all these which we have read over here. So whatever questions will be there, you will be able to solve it with respect to your other knowledges, okay, knowledge which you have created over here. Somatic hybridization, let us discuss one important topic which you should cover end to end. Somatic hybridization. Yes. Right, this is nothing but somatic hybridization. Somatic Hybridization. What do you understand by somatic hybridization?
The protoplast fusion.
Somatic hybridization means, again, a definition if I ask you.
So production of hybrid through the fusion of protoplast of two different plant species.
Right.
Particularly in sexually incompatible plants.
Okay, good. Over here, somatic hybridization means over here we are not going with respect to the you can say that gametic intercross we are not going ahead with. Over here we are doing the fusion of two parent or two plant species by the protoplasts. So it is a technique which is used in plant biotechnology, where protoplast from two different species are fused to form a hybrid cell, which can then regenerate into a whole new plant. So two incompatible plant also can be done over here, can be fused together over here with this particular process of somatic hybridization, which you wanted to say. So this is one particular definition which you will be getting over here with respect to somatic hybridization. Now, the question which can come in the examination, which has come also, what are the techniques of somatic hybridization? What are the techniques, guys? How you will be doing somatic hybridization, or what is the procedure of somatic hybridization? Tell me.
Spontaneous fusion.
First, what you will be doing?
Isolation.
Doing isolation.
Procedure, actually.
Second, what you will be doing?
Fusion.
Fusion of protoplast. How you will be doing the fusion of protoplast? How you will be doing the fusion of protoplast? One, you can go ahead with chemical.
Sir, spontaneous fusion, mechanical fusion.
Mechanical and induced.
Right. Chemical में क्या लिखोगे आप? Example बताओ for the exam directly.
PG,
NaNO3 का use करके PEG.
Great. PEG you can utilize, on one or more chemical also you can write over here. Chemical or electrofusion also you can go ahead with the help of electricity. Because over here we will be
lysine also you can.
Right. We will be giving them high voltage pulse and they might be fusing because of this. ठीक है. फ्यूजन में आपका आता है स्पॉन्टेनियस एंड इंड्यूस्ड फ्यूजन ऑल्सो. देखो स्पॉन्टेनियस फ्यूजन, सॉरी, हाँ, इसको आप ऐसे रख सकते हो पहले. देखो आइसोलेशन में हाउ यू विल बी डूइंग द आइसोलेशन. लेट अस टॉक फर्स्ट अबाउट दिस आइसोलेशन. हाउ यू विल बी डूइंग
मैकेनिकल और एंजाइमेटिकल.
राइट, देयर विल बी केमिकल and mechanical. And chemical, what you will be utilizing?
Sir, proteinase or cellulase.
Cellulase, pectinase, cutilase, that you can utilize over here. In mechanical, physically it will be disrupted, right? But this is less efficient, so that's why this is not utilized much. Now you will be doing the fusion of these protoplasts which you have taken out. How you will be doing the fusion over here? One is spontaneous, another one is induced. Spontaneous is it will be happening naturally. Induced means you will be inducing two protoplasts to fuse together. So in this you will be finding chemical method, PEG, calcium ice, sodium nitrate you can utilize. Or mechanical method you will be finding over here, that is micro manipulation you can do under the microscope. And third is
fusion micropipette.
Right, electrofusion also you can do over here with respect to this. Then the third technique which will be coming over here, or the third procedure will be
selection of hybrids.
Selection will be done over here. What are the techniques of doing selection?
First, visual.
Techniques of doing selection of hybrid.
First, visual selection.
Visual selection.
Yes.
Apart from that, visual तो करोगे ही ना, उसके अलावा क्या करोगे? For example, what is the problem over here with respect to this selection? Problem क्या selection में? Why we will be doing the selection over here? Because either we can get over here non-fused parents or non-fused progeny. We can get the homokaryons. Homokaryons means the fusion of the same species. It might happen the fusion of the same species has happened. This also we don't want. We want only one thing, that is heterokaryons.
Heterokaryon.
Also the functioning heterokaryons. These are nothing just words, guys. Heterokaryons are the hybrids which are desired over here. That's why we want to get ahead in selection. And selection visual तो होगा ही होगा, visual तो करना ही पड़ेगा, but there are certain methods, techniques with respect to selections. What are the techniques? One technique name which is used by the academicians is complementation technique. लिखना है exam में direct. Complementation technique is used when both parents have deficient traits which complement in fusion. These both are having deficient traits. For example, one is lacking chlorophyll and other is lacking nutrient synthesis. Then you can utilize this particular function, because the hybrid will show both these particular part. And you can understand the difference between the homokaryons or the heterokaryons based upon complementation technique. The other techniques which is utilized over here is a metabolic inhibition. Fluorescent labeling you can do. This is the much better method, fluorescent labeling, which is being utilized by these particular people. Morphology.
MI में ये I क्या है सर? Metabolic
inhibitors. मैं अभी आपको सब कुछ दिखेगा इसमें, don't worry. I'm just doing some brainstorming with you all guys. Otherwise every damn thing is written in this PPT. ठीक है, आपको कहीं नहीं जाना.
draw resistant.
हाँ ठीक है, तो ये हमारा हुआ थर्ड सिलेक्शन कर लिया हमने. अब फोर्थ व्हाट विल बी डूइंग आफ्टर द सिलेक्शन?
regeneration या culture?
Culture we will be doing. First of all, we will be now doing the cell wall regeneration, first cell division, and then we will be going into the organogenesis. That is nothing but your regeneration of whole plant, organogenesis, embryogenesis. Now let me tell you over here, isolation of protoplasm. आराम से पढ़ो, इसको short में बना लेना. Chemical method, mechanical. Fusion you will be doing spontaneous, induced chemical, mechanical, and electrical. Selection will be of parental, homokaryons, heterokaryons. Selection methods can be utilized complementation technique, metabolic inhibitors, fluorescent labeling, morphological or physiological traits. And then you will be doing the culture and regeneration into whole plants. This is nothing but your somatic hybridization. Understanding, guys? ये एक बार करना है, just make the short form of it and you are good to go. Again, I want you guys, those who are like attending these sessions throughout, like some of you have been attending since the starting, and I know most of you are very close to your selection also. Let me tell you, just these 3 months, just try to devote wholeheartedly. Okay, wholeheartedly you try to devote on means, because this is a time period when most of the people get worried about prelims. And when you will be near to prelims, you will be worried about your means, thinking that what if you will be clearing the prelims? Means to prepare hai. Again, you will be getting into this negative cycle. Okay? Those who are in their first and second attempt, try to take precautionary measures. Those in their third and fourth attempt, they already know it. Okay? So try to be wise in this time frame. If you want to utilize me or with respect to anything, you can utilize, right? So what I was telling you, the application part. What is the application of somatic hybridization, guys? Directly exam में यही सब पूछते हैं, nothing else. Application of somatic hybridization.
Production of novel varieties.
Okay, right. Novel traits you can introduce over here. Good. ठीक है, novel crop production can happen. Good. Apart from it,
overcome cross incompatibility.
Overcoming sexual incompatibility. Those two species which are not sexually compatible, we can do it by somatic hybridization. And this is basically utilized in a lot of horticultural crops. Then transfer. Tell me more.
Production of fertile diploids and polyploids from sexually incompatible crops.
ठीक है, हो गया ये वाला point. Apart from it,
so production of allotetraploid plants.
Production of
allotetraploid plants.
Allotetraploid, right? ठीक है.
If we go for cytoplasm transfer, then it will, after 1 year, it will be destroyed. But in case of this type of Backcross,
hybrid also you can say.
Yes, sir.
Cytoplasmic hybrid production you can go ahead with. ठीक है, good point. ऐसे point बोलो जो एग्जामिनर को भी लगे कि पढ़कर आए हो. It should not be random or generic points. Right? So hybrid
Sir,
you can create over here. Transfer of disease.
organellar genomes. So,
ठीक है, तो यहाँ देखो, ओवरकमिंग सेक्सुअल इनकॉम्पेटिबिलिटी, एग्जाम्पल डिजीज एंड रेजिस्टेंट साइटोप्लाज्मिक प्रोडक्शन, combining polygenic trait, novel crop production, which you were saying, preservation of elite germplasm, genetic studies and mapping. ये तो पॉइंट आपको हर जगह ही दिखेगा. कुछ ना समझ में आ रहा हो, genetic studies and mapping you will be finding. But these are certain things you are going to find with respect to this. Now, what are the disadvantages? You will be finding that this is not a very reliable method, this somatic hybridization. So there are many limitations and disadvantages also, and that's why it is being asked in the examination too. Tell me, what are the disadvantages and limitation with respect to this?
सर फर्स्ट तो रीजनरेशन केवल वहीं पे पॉसिबल है जहाँ पे प्रोटोप्लाज्म फ्यूजन हो सकता है.
ग्रेट. तो यहाँ पर रीजनरेशन डिफिकल्टी है भैया बहुत ज्यादा.
sir, non-viability of fused products.
That means what do you want to say, that low hybrid recovery rate? हाइब्रिड ही यहाँ पर नहीं बनते. एंड द सेम गोज फॉर इन विट्रो फर्टिलाइजेशन, आईवीएफ टेक्निक में भी जो आपको देखने को मिलता है इन ह्यूमन केस.
So genetic instability also.
जेनेटिक
हाइब्रिड्स
इंस्टेबिलिटी,
यूजुअली स्टेराइल होते हैं.
राइट. लैक ऑफ डिजायर्ड ट्रेड एक्सप्रेशन, हाइब्रिड स्टेरिलिटी यू विल बी गेटिंग. तो सारे पॉइंट यहाँ पर लिखे हैं. रीजनरेशन डिफिकल्टी, लो हाइब्रिड रिकवरी रेट, जेनेटिक इंस्टेबिलिटी, लैक ऑफ डिजायर्ड ट्रेड एक्सप्रेशन, hybrid sterility. Now your job is to just compile it and keep the keywords over here and keep revising it once. Okay, once before your mains. Then you will be doing it in mains. Don't worry about that. We will be again having this series during the mains also. There we will be revising. But I want you, all of you, to give a mains at that point of time, guys. ठीक है, तभी मजा आएगा. ठीक है, disadvantages you have known over here. Now there is one more question which has come with respect to soma clonal variation. What do you understand by this soma clonal variation?
So that is genetic variation produced due to that plant tissue culture.
Good. That means due to this tissue culture, there is something genetic or epigenetic changes observed because of this tissue culture. Now these variation can be heritable only or non-heritable also. Can be heritable also, non-heritable over here.
No.
It might be beneficial, neutral or harmful too. And it is a natural byproduct of this tissue culture. ठीक है, तो सोमा क्लोनल वेरिएशन रेफर, देखो ये लिखा नहीं गया, दिस इज नॉट रिटन इन योर सिलेबस लाइन, बट द क्वेश्चन हैज बीन आस्क्ड ट्वाइस. दैट मीन्स इट बिकम्स इंपॉर्टेंट. So over here, soma clonal variation refers to the genetic and epigenetic changes. Variation can be heritable, non-heritable, and this you can see over here. What are the causes, if I ask you, of this soma clonal variation? Causes of soma clonal variation.
Sir, that is regeneration system.
First of all, physiological causes you will be finding.
Yes.
Second, you will be finding genetic causes. Third, you will be finding over here, or genetic causes. Third, biochemical causes.
Yes.
In physiological causes, what you will be finding, guys? Physiology में हो सकता है that we give excessive amount of growth hormone regulators, high concentration of auxins or cytokinins we have given. There might be the condition in cultures, light, temperature, culture duration. These are nothing but physiological, right? So this might be the problem. Genetic में what you can find? The problem with chromosome number, problem with chromosome structure, deletion, duplication, inversion, translocation. And what will be under biochemical? Loss of photosynthetic ability. Antibiotic, herbicide resistant can happen over here. So causes: physiological, exposure of growth regulators, culture conditions. It can be genetic, like change in chromosome number, aneuploidy, polyploidy, chromosome structure, duplication, inversion, translocation. Already we have discussed everything. Gene mutation, point mutation, frame shift mutation can happen. ठीक है, biochemical causes: loss of photosynthetic ability, antibiotic herbicide resistance, altered enzymic activities also over here. This can also happen over here. Right, important to learn what are the causes as it is score lakh lo. This will be helpful for you. The third question which can come over here is the application part of it. Application of soma clonal variation, guys. Tell me the application of soma clonal variation. बताओ भाई.
So genetic improvement.
Right, one, but basics now you can add it everywhere. That is crop improvement, genetic improvement we want to do. Next. Disease resistance, pest resistance, abiotic stress tolerance, quality traits improvement. Right, everything which we have discussed. ये तो एक selection method ही है ना. Ultimate objective तो हमारा same ही है. Crop improvement, breeding, disease resistance over here, abiotic stress tolerance over here, improvement of quality traits, herbicides and antibiotic resistance, CMS, shortening breeding cycles. So these are all points which are applications of soma clonal variation you will be finding. Okay. Now, this is all for this particular class which we have discussed over here. Okay, major portion I have finished over here. Rest of the parts you can see from the lectures, but majorly we have covered this particular part over here. Okay, kuch new tha, something I gave you extra from which I have not discussed in the classes. This particular part we have completed. Check out the PPT which I have given you. Application, this you will be doing. Molecular markers is over. Pure line is over. Modes is over. History of plant breeding we have seen. Heterosis, somatic. This we will be again seeing in unit number 6. Yeah, role of interspecific and intergeneric. 1 or 2 questions have come till now. Just see it from the PPT or my class lectures, right? And these two we have discussed over here, but I have not discussed in my class. Okay. एक यहाँ पर आप एक वो चेक करना दैट धारा हाइब्रिड मस्टर्ड में चेक करूंगा. आई माइट बी कंफ्यूज्ड ओवर हियर विद रिस्पेक्ट टू दैट. व्हाट वाज द क्वेश्चन which you have like Shashank, you were saying something.
Sir, DMH 11 is self-pollinated or not?
ठीक है, एक बार मैं इस पे जरा रिसर्च करूंगा, है ना. देन आई विल बी कमिंग टू यू गाइस, डोंट वरी अबाउट इट. ठीक है, अपार्ट फ्रॉम दैट द कॉन्सेप्ट शुड बी क्रिस्टल क्लियर एंड दैट इज ऑल फॉर दिस पर्टिकुलर क्लास. अगर आपके कोई डाउट है, इफ यू हैव एनी डाउट यू कैन आस्क, अदरवाइज वी आर एंडिंग द क्लास गाइस. चलो ठीक है, फिर वी विल बी एंडिंग. दैट्स रियली ग्रेट फॉर यू टू सिट ओवर हियर. नेक्स्ट सेशन में मिलेंगे हम लोग. ठीक है, ऑल द बेस्ट गाइस.
थैंक यू सर.
थैंक यू सो मच.
Thank you, sir. Thank you, sir.
Thank you, sir.
थैंक यू.